Hello?
Hello, good morning, Dr. Andre.
Hey, okay, good. You can see and hear me, I suppose, right?
Yes.
Perfect.
Okay.
Very,
Just
very
15 minutes and start, inshallah. Okay?
Yeah, no problem. Do you want me to test the screen sharing too or no?
Okay.
Okay,
Yes,
hold on
please.
a second. Give me one moment. I think what I will do, this one and this and this. Okay, hold on. Just 1 second.
Okay, take your time, please.
Put that here. And I will screen.
I think because we are just sharing our official poster. Yes, that's why. Yeah, just 15 minutes. We have an opening ceremony video.
Yes. Yeah.
After that, we will introduce you and we will start the first session with you, inshallah.
Yeah, that's perfect.
Okay. Thank you very much. You are welcome.
Okay.
Thank you.
Okay, good. Thanks. I'll be here on mute and turn my video off.
Okay. Thank you very much.
Hello, Assalamu alaikum. Good morning. Dr. Hanan, you will chair the session now. Hello, Dr.
Yes, please. Hello, doctor. Yes, please. We will start now, inshallah.
Okay. Thank you.
Assalamu alaikum wa rahmatullah. It's my great pleasure to welcome you all to the second international webinar on science, sustainable development and ecosystem in Saudi Arabia. My name is Hanan Al-Hashir, and I'm an assistant professor of plant ecology at the Biology of Department, College of Science, Imam Abdurrahman bin Faisal University. Again? Faisal? No? Can anyone hear me?
Yes, I can hear you very well.
Okay, thank you, Dr. André. So we welcome you to the second international webinar on science, sustainable development and ecosystem in Saudi Arabia. My name is Hanan Al-Mahashir, and I will be the moderator of today's sessions, session number 1 and session number 2. At the end of each presentation, we will have 5 minutes for discussion. So if you have any questions, just write them down in the chat box below, and we will address them at the end of the session. We will also have a small questionnaire just to type your name and your opinion about the session, and we will send then a certificate of attendance. We will start with our first speaker. The principal speaker of today's first session is Dr. André Schleifer. He is a full professor at the University of Illinois. He received the NSF Career Award and the ONR Award and was an AS Doctoral New Investigator. André is an editor for a journal and actively organizing national and international schools, workshop and tutorials to advance the community around the cutting-edge first principle simulation of material science. His title today is Engineering the Defect Mobility, Modern Materials for Batteries and Quantum Computing. Dr. André, we
Great.
welcome you.
Thank you, Hannah, so much for the introduction. Before I say much, let me try to share my screen. I think
take your time.
This one and share. And what I'll do is I'll start the presentation also. Just 1 second. So can you see the slide and the correct screen?
Yes.
Perfect. Okay, great. So then thanks again for the nice introduction. It's my pleasure to present at this webinar. And it's unfortunate that I cannot be in person at the webinar, because as you see, online discussions it's never as interactive, and it's always very difficult to get real collaboration started and things like that. But nevertheless, it's my pleasure to be here and kick off your Monday morning session. And what I want to do over the next few minutes is I will tell you about my group's research in computational material science, right? So we're a simulation group, and I will tell you a little bit about our work on engineering defect mobility, or what we're envisioning there. You can see here, and maybe I will turn this into a laser pointer like this. You can see here my research group. This photo is taken last fall. Right now we have a lot more snow in Illinois, so that would be another reason to visit Saudi Arabia this time of the year. But nevertheless, these are the people who actually do the work. And what I also want to do is I want to acknowledge funding agencies and students. So the results that you will see today are largely based on research done by Anish and Sophie, as well as Alina and Yifan. I will tell you that later in the talk again. And funding agencies are listed here as well, NSF and ONR, and then various, the DFG is contributing as well. And then we use computer time at both, the Blue Waters Computer and at Argonne National Lab. Okay, so with that, let me start my talk. And because I'm the first speaker in this session, I wanted to put the work that I will be discussing a little bit more into a sort of a general context. And one thing that, of course, maybe many of us, if not all of us, are always claiming in our introductions of papers and in abstracts of talks and all these kind of things, is that many challenges that our society is nowadays facing, our global society is nowadays facing, can be either solved or benefit from advanced materials. Okay, so that includes, of course, maybe energy and climate change. So I like to show this diagram here. Many of you have probably seen this many times, which plots the either the average temperature or the carbon dioxide concentration in the atmosphere since 1880 over the last, let's say, 150 or 130 years or something like that. And we see this increase, right? So we claim that materials can help solving that. Other examples are health and medicine, transportation and water, and food, similar to the topics of this webinar. And one example that I want to actually show you guys is this actually the case, right? So where are the real materials problems? And so what I pick here is the example of photovoltaics, right? So that's an example that many people are very familiar with. And it is, more importantly, an example where we actually have a good idea what the maximum efficiency is that we can achieve in a solar cell. Now, when I say solar cell here, I mean a single junction direct bandgap semiconductor, right? So these are, of course, some constraints, but for that we have the Shockley-Queisel limit, right? Anyone who works in solar cells has probably heard of that. It essentially determines the maximum theoretically achievable efficiency of that device as a function of the bandgap of the absorber material. Okay, the bandgap, in electron volt is then the x-axis. So that's a materials parameter. So with that, we can look at this efficiency curve, and we can immediately say, okay, well, if we're at the bandgap maybe around, like, I don't know, 1.5 or something, or slightly below, we get very good efficiency. That's fine. But it also tells us is if we have a given solar cell material that we're currently using, like for instance silicon, and we know that we achieve with that material, an efficiency of 29%, it tells us that it's not that much more that we can improve, right? Because we're already at 29%. The theoretical maximum is like, I don't know, 33%. So that's not that much, right? And what that can do is it can guide our research efforts, right? It can decide, and it can help us decide what should we actually be researching. Now, this example is sort of intuitive in a sense that for these single junction solar cells, we have this number, we know this analytical expression that gives us this curve, and we can look at this data directly. I want to put this a bit more into contrast. So what we also have is, in many other instances, like the examples I listed earlier, maybe energy storage, for instance, we don't have such relations, right? If you want to know what's the best possible efficiency or energy density, let's say, of a battery material, we don't know necessarily a similar relation like the Shockley-Queisser limit. But what we do have nowadays is materials databases. And that's the case I'm trying to make here. We have large databases on the internet, openly available, not all of them, but some of them. They contain a large number of materials data and a large number of materials parameters, right? Both theoretically and experimentally. And that is a powerful tool because in cases where we do not have a Shockley-Queisser limit, we could use that data that we have already stored in the internet and train, for instance, machine learning models on that data. Now, we all know machine learning models may not be super great at extrapolating, and that does not maybe give us the final answer in all instances. It depends very much on the quality of the data. All that is true, but one thing that it can do is, once we have that model, we can run predictions again for this large number of materials in the database. And we can compare, then we can plot that, right? We histogram it in this case. We look at how many candidates do we have that have a certain power conversion efficiency. So the example here is not a single junction solar cell anymore. Maybe this is a multi-junction solar cell, okay? For which the Shockley-Queisser limit doesn't work like that because that was only for a single junction cell. But we can then histogram the data from that machine learned model. We see where is our currently best candidate in relation maybe to the theoretical. So now that's not a theoretical maximum anymore. It's sort of a practical maximum, and I'm putting quotes here even because it's really just the maximum that we computed for this data. But what this data nevertheless can tell us is it gives us an idea of what can be achieved more or less with this, given paradigm. So for instance, in this case of this multi-junction cell, maybe we can get efficiencies realistically that are a little bit higher, up to maybe 50% or something like that. There are a few materials out here at very high efficiencies. It gives us an idea of what's possible, right? And it can guide how much is the problem I'm investigating actually a materials problem. How much can we gain? It also can serve as an estimate of what is realistic as a near-term improvement. So let's say if your current example is already up here somewhere, well, then what is realistically achievable, right? I mean, not that much on this scale. And yes, you can say, okay, this is not all the materials that are theoretically possible. That's true. But how likely is it that within 10 to 15 years you come up with this much, much, much better candidate? So it gives us an idea of how much of a materials problem is this that we're actually trying to solve. And it can also help us to say, okay, if in the Shockley-Queisser case, let me just go back to that, we're maybe at the 30% maximum already with silicon, what that tells us is we need to go beyond what the Shockley-Queisser limit assumes, right? For instance, a multi-junction solar cell. So that is that can, in other words, tell us what are interesting questions that we can ask material cells. And so with that, I want to just conclude the solar cell example, because I think actually for the solar cells, there are a lot more severe challenges that are of practical nature. Not so much we need a better absorber material in a single junction solar cell. Those are related to things like deployment, right? What we want is, we want ideally large-scale adoption of these solar cells, right? Not just a few households installing solar roofs and things like that. That's not going to help with climate change, right? We need a much more large-scale deployment. And thinking about that is probably more important because we have silicon ready to go, right? That's the message that I want to send. We don't have to wait for a better material that's maybe a little bit better in efficiency. We just make silicon. It's cheap. We have it. We can make it in large amounts. The other thing is, assuming we do that, right? Let's say we build solar farms. The problem all of a sudden becomes at night, those don't work, right? And then storage becomes an issue. So we need then large-scale storage. And maybe that is much more of a materials problem. I haven't done any of these analyses for storage materials, but that would be something to look at. And then the last thing is timescale, right? If we wait for a material to be, if we wait for a much better material to appear, or even a much better paradigm to appear, and we wait another 10 to 15 years, well, those 10 to 15 years are important, right? There are analyses in the literature that say that even if we were to stop emitting CO2 today, we're not going to prevent climate change, right? So that, and of course, we cannot stop emitting CO2 today, right? Even if we replace everything with solar cells, that there are a lot of other sources of CO2 emissions. And so, in other words, the sooner the better, right? And so if we start doing those kind of things, that may be more important. And how can we tackle those problems? Well, not necessarily with material science, right? So this could include policies, right? Maybe we have to mandate that power generating companies have to have a certain fraction, and maybe a large fraction of their power generated from renewable energy. And then we have to maybe look at storage and potentially even CO2 capture materials. Anyway, what I wanted to use these general slides in the beginning for is just to give you guys the idea that anytime we're looking at a materials problem, it is valuable to use the data that is available nowadays in large amounts, and in many cases freely, to sort of assess, not for the majority of the project, but to sort of assess to what extent is the problem I'm solving actually a materials problem, and then maybe pursue it or not. And that, when we talk about interesting directions, I think in the bio realm, there is a lot of, in medical materials, there is a lot of cases where the underlying problem is actually a materials problem, and it's worthwhile looking at. And in the particular instance of this talk, and that's what I'll now start when I talk about some actual science and results, I believe in quantum computing, it's a similar situation. I think there we don't have the silicon analogon that everyone can simply use and scale up, right? I think there are a lot more materials problems to be solved, and that's part of the reason why we started looking into this. Okay, so with that, let me show you at least one motivation slide for what the title topic of my talk is, and that will be mostly actually the quantum computing materials, not so much the batteries. I'll give you one example for that. But what we started looking into is to what extent can we affect atomic geometries and defects in a material through electronic excitations. And so I like to show this cartoon picture because it gives you, the audience and you guys, an idea about time and length scales I'm thinking of. So we will look at atomistic defects, single atom point defects, right? Individual atoms being moved around. And we are looking at time scales that you can kind of see here, maybe, in the range of a couple of hundred of picoseconds. And the processes that I'm thinking of in this talk is related to photo excitation. So where we take a material, this is now the valence band and the conduction band, a material from its ground state into an excited state by exciting it, either with an ion beam or with a laser pulse. In the talk, I will focus on ion beams, actually. But in any case, we start with an initial excited electronic state, and then we sort of understand the time evolution of this excited state. And the case I will make is that the excited state itself can affect your defect properties and affects things on this atomic defect scale, and that's the interesting part. Now, for batteries, since I put it in the title, I want to at least give you an idea of what I had in mind there when I put it, even though the results will be more on the quantum computing side. So batteries nowadays, some people think about photo-accelerated batteries, which are essentially batteries that you still charge traditionally by connecting them to wires, but by illuminating them with light simultaneously, you can accelerate this charging process, right? Why is that interesting? Well, if you think of, I don't know, electric cars, do you want to sit at a gas station or a battery charging station for 2 hours or for 2 minutes, right? So that if you can accelerate this charging process, for instance, by light illumination, because the light makes the ions in the battery more mobile, that would be very desirable. So that is something that people research. I pulled this literature reference here. What I will focus on this talk is more creation of defects in a material and maybe manipulation of defects. And so by that, this example here is for molybdenum disulfide monolayers, where ion beams are used, in this case highly charged ions, to create those kind of defects. Now, this is maybe a bit larger than a point defect. In the presentation, I'm more thinking of maybe point defects. So ion beams can be used to create those defects in materials, and those defects are then potentially interesting for instance, for qubits, single photon emitters, and things like that. In addition to manipulation of the material through those beams, some applications include characterization of the material by beams. So the example I like to show here is helium microscopy, where you use a helium ion beam, you expose a sample to that ion beam, and as a result, secondary electrons are ejected from the material, and those are detected. Okay. those are detected and used for imaging. So we will look at those secondary electrons. So in the following, so I guess if the very high level view of my talk is that we want to understand the connection of excited electronic states induced by an ion beam and the resulting dynamics of ions in the material. Okay, and specifically I will discuss 3 points. One is the initial energy deposition of the ion beam in the material, then the secondary electron emission, because we will talk about low dimensional materials, as you will see in a second. So there will be secondary electron emission into vacuum, and forces that determine the defects in the material that then result, for instance, these guys here. Okay, so I will not bombard you with equations, but I want to show at least the framework that we're using, and that is the framework of time-dependent density functional theory, where we time propagate these Cohen-Sham equations. So if you've ever done density functional theory, this is the time-dependent version, and our ion will be in this potential. Okay, we're using the Q-ball code. Again, I will not discuss all these details, but this code is well suited for these applications because it's very well parallelized. Okay, so now, in order to set the stage for what you will see in the following, I want to show you one short movie here, which illustrates the kind of the simulation output we're getting. So what you see here is aluminum, a thin layer. In this case, we have 4 layers of aluminum here. These balls are the aluminum atoms, and we'll shoot a proton through this material coming from the left. So here's the proton, moves along this trajectory, initially completely charged. Now it hits the material, it excites electrons, as you can see here from these colorful things, and then it leaves the material with some electrons around it. Okay, so this process, as it moves through the material, this is when it deposits energy, and when it leaves the material, we didn't see the secondary electrons. I will illustrate that on the next slide, but there will be secondary electrons emitted into the vacuum here, and the particle itself, the ion itself, takes electrons with it. If you want to read about this in slower speed, so to say, I put the reference here on also on the next slides. So in other words, the cartoon picture is that we have a certain sample that we expose to a particle beam. Energy is deposited initially. This is called stopping power. I'll tell you that in a moment. And then in addition, electrons are emitted from the material, and forces result as a consequence of these electronic excitations and the electron emission. Okay, and that's what we want to model. So what you can see at the bottom is now a snapshot from one of our simulations. This is sort of a top-down view. You can again see here the aluminum, and in the background you can see these colorful areas here. This is electron density. Okay, dark means high electron density. So the aluminum, of course, is very dark here because the majority of the electrons is of course still in the aluminum, but you can see some electrons are around this proton now, and then this bluish greenish area here is secondary electrons. Okay, and then what Alina did is she developed this very nice analysis where she understood the electrons around the proton after it leaves the material. In what state are those electrons? So some of them are 1s, you can see this here. Some of them are 2s and 2p electrons, you can see that here. So she can really classify what states do these electrons that the proton captured occupy. And she also came up with analysis of secondary electron emission. And so what you can see here is a plot of the number of electrons as a function of position, sort of perpendicular to the material. Again, in the material, of course, there's the majority of the electrons, but you can see here, color code is as time in this case. You can see that initially, which is dark blue, there is no electrons here in the vacuum, and then as the proton moves, secondary electrons are emitted. So these tails here are secondary electrons. And then we integrate this, we plot it, and we analyze it. And the first thing that we found is that as a result of, so our simulations are using periodic boundary conditions, and that makes them dependent on how much vacuum we explicitly include. And you can see these decreases here. We think this is finite size effects. And so we have to use absorbing boundaries. I'll do that in a second for graphing. Okay, now let's start with the energy deposition. Okay, so what happens initially, we're coming in with a proton, and we're studying how much energy does this proton lose. This is this delta E as a function of position in the material. Okay, so dE over dZ is what we call stopping power. It's the friction force that the proton experiences. That is the quantity we're visualizing here on the y-axis. And we do that as a function of the kinetic energy of the projectile. And so what you see here is, let's just compare the orange curve, which is bulk aluminum, and the green curve, which is our thin aluminum. And we found that more or less independent of the precise simulation parameters, so we made sure that those are all well converged, we find that in the sheet, right, we always get higher stopping, meaning more energy is deposited in the sheet. And we try to explain this thickness. And what Alina came up with is essentially 3 candidates for what the reason is. One is the charge of the projectile, of the incoming ion. One is a certain polarization effect in your material. And the last one is surface plasmons. And without walking you through all the analysis that she did, as I said, if you want, you can take a look at this in this paper. She determined that surface plasmons is indeed the difference between bulk and the sheet that give us an additional channel for depositing energy. And the way she found that is that she looked at the dipole moment in our simulation, and she plotted that as a function of time. And for different thicknesses of aluminum, in this case 4, 6, and 8 layers, she found this oscillatory behavior of the dipole moment. And that's a clear indication of plasma oscillations. Now, how do we know it's surface plasmons? That's a good question. And the way she analyzed that is by looking at the Fourier transform. And she plots here again the dipole moment now as a function of frequency, and we find that especially for the thin layer, for the thinner aluminum, where in other words there's a lot of surface compared to the number of atoms, the dipole moment is actually much closer to the surface plasma frequency of aluminum than to the bulk plasma frequency. And for thicker layers, we get kind of closer to the bulk. So in other words, she showed that in thin materials, 2-dimensional materials, or thin materials, surface plasmons need to be taken into account because they are an additional energy dissipation channel for the ion beam. Okay. She also looked at secondary electrons. I'll keep that somewhat short because I want to focus a little bit more on that for the graphene, but I'll tell you nevertheless the results. So what she plots here is how many electrons did we emit as a function of thickness, and she finds that consistently the exit side emission, meaning in the movie it was the right-hand side of the material that you saw. In other words, where the projectile exits, we see consistently more secondary electrons emitted there. There is secondary electron emission also on the entrance side. And then she fitted that to a model to determine both the secondary electron yield, which is gamma. That is a quantity that's measured in experiment. And if we compare the entrance side yield, for instance, with experiment, we find relatively good agreement. So I think these simulations are predictive for that. And then she can also extract the escape depth. And she finds that the exit side has both a higher yield and a deeper escape depth. Okay, with that, let me come actually to graphene, because one obvious criticism of what you've seen so far would be, well, aluminum is a nice example, but it doesn't exist in a monolayer or something like that. That's true. And so this was just our first test case. We then started looking at graphene, and the movie in this case just illustrates what graphene looks like and what the electron density in graphene looks like if you were to scan it. For graphene, what Alina did is she looked at the spatial dependence of the energy deposition. So the spatial dependence of the energy deposition correlates with the distribution of electrons in your graphene material. She explained that. That is in the first paper. I will discuss that not so much detail. She also looked at comparing this to larger scale simulations together with collaborators, in this case Henrike. And then she found that in graphene, because that's a very good conductor, the damage is actually relatively light because electron conduction is fast, and so it can equilibrate fast. I'll show you that on the last results slide in just a second. Okay, so let's look at graphene. Again, the references, you have them here, and then partly throughout the next couple of slides. The first thing that Alina looked at is, just like before, she looked at how many electrons does the projectile capture as it goes through the material. And now she looks at different trajectories here. So this is a top-down view of a graphene hexagon, continues, in plane. And the letters label different perpendicular trajectories through the cell, right, through the graphene layer. And in particular, we looked at a channeling case and a centroid case. The centroid case we picked because it was studied in the literature. So we have data to compare to, and we find good agreement. The channeling case we show here in this plot because it shows this interesting peak behavior here, or oscillatory behavior almost, as a function of proton velocity. And she attributed that to the particular electron distribution in graphene, right? So there are sigma and pi electrons that contribute at 0.4 and 0.6 atomic units of velocity, which is roughly where we see this peak and this dip. So we hope that this is something that should be measurable also in experiment, and it can tell us the charge state as a function of, projectile velocity. In this case, we showed a proton and trajectory. Then, just like before, for aluminum, Alina analyzed the secondary electron, right? The number of emitted electrons. And just as before, we find much more electrons are emitted on the exit side. You can see the red curve is higher than the orange curve. And she also finds that the velocity dependence of exit side and entrance side is different, right? So in particular, the exit side emission seems to have this maximum here at 50 kilo electron volt. You may ask, okay, why is that important? We believe that this has implications for helium microscopy. If in a helium microscope you're trying to optimize your beam kinetic energy to get the most secondary electrons out for good image contrast, these curves can tell you where such a maximum would be. And it can also distinguish between exit side and entrance side. We also see that there seems to be a threshold here at low velocities that correlates with the work function. Now, let me talk a little bit more about the secondary electrons before I come to the forces. One thing that we struggled with is, and you saw this before for the aluminum, finite size effects. And so for graphene, that situation is similar. But before we look at how we can fix this, we find that the number of secondary electrons does not just depend on exit side and entrance side. It also depends on where we impact. So you can see here different trajectories compared. And it depends on the projectile velocity, which is this color code here in atomic units again. And we can see that the number of secondary electrons that are emitted depends also on the velocity. Now, the problem that we still encountered are these finite size effects. And in order to fix that, Yifan now implemented absorbing boundary conditions into our code, which prevents this merging of the electron densities because of periodic boundary conditions. And with that, we were able to actually look at the pulse shape of the secondary electron pulse. This is the second to last slide. I'm almost done with this. So we look at the pulse shape of the secondary electron pulse. Again, on entrance and exit side, we find, just as before, exit side emission is much stronger, and it's also much more sensitive to beam parameters. And in particular, one thing I want to point out is we find an opposite trend with the velocity. Okay, so let's just look at the trajectory A here, and the velocity increase is going from blue or orange to green. So blue, orange, green leads to less emission on the entrance side. But if we go here from blue to orange and green, it actually leads to more emission on the exit side. So the velocity trend is completely inverted between the exit side and entrance side. That's something we're currently trying to explain. We also see that the pulses are overall on the order of a femtosecond, right? So they're sort of confined within a femtosecond. And we always get higher current for higher velocity on the exit side. And then the last thing I want to point out is there is a double peak here for certain velocities and certain trajectories. That's some feature that we're also currently trying to understand. We believe it may have to do with some resonance with capturing the electrons near the projectile, but that's preliminary data, and I don't want to claim that just yet. All right, before I conclude, one last slide of results. You may now ask, okay, how do I actually really use these ion beams to create defects in the material and maybe tweak these defects? And for that, I want to show you this last slide here, in which case we use silicon ions as our projectile. And what you see here is both for the centroid trajectory, and the only difference between left and right side of the figure is the charge state of the silicon ion. Okay, so we start with a 4+ ion. You can see that on a time scale of between 0.02 femtoseconds up to 0.6 femtoseconds, that initial perturbation is completely dissipated if your charge state is low. That is not the case if you use a highly charged ion like the silicon +12. Right? After 1.5 femtoseconds, there is still a significant electron density perturbation here, simply because this higher charged ion creates a lot more electronic excitations in the first place. Okay, so then that can again tell us what beam parameters do we need to use to create forces, right? So large perturbation of electron density leads to large forces on the ions and subsequently creates defects in the material. With that, I would like to conclude my talk so that we have a little bit of time for questions. And what I said is I'll summarize just 3 results. One is that for aluminum, we found that surface plasmons contribute to the energy deposition and need to be taken into account and studied in when you describe these kind of materials. We also were able to really look at the pulse shape and the pulse behavior of secondary electron pulses. We can maybe optimize image contrast and things like that. And we were able to tell how the charge state of a defect influences the electronic excitation. So that's, I think, were specific examples of how we're envisioning using ion beams to manipulate materials. And just again, as the first speaker, I want to also give maybe a bigger picture view. And so to summarize my first part of the talk, I want to say that one thing we should all do is we should pick high impact materials problems, right? We should use all available data that we have to assess where is a real material problem that needs to be solved. I think examples include CO2 capture, bio recycling. So this webinar is about sustainability. I think that's a great example where a lot of materials problems exist. Maybe batteries and maybe quantum computing. The other side of this is we need diverse and interdisciplinary teams, right? Not just the material scientists maybe looking at their own little problem, but talking into with teams of larger dimension and across departments and across disciplines, right? Including policymakers, because I think a lot of these problems that we're facing in society are not necessarily solved on a just scientific problem, but we need to also then come up with policies, and policymakers need to inform the scientists what they need to know to come up with efficient policies. We need, and that's something that all of us can do, and that's why I want to say it, we need open exchange of ideas, and in particular, all of us should upload their data that they're generating, maybe their workflows of analyzing this data, the scripts, the Jupyter notebooks, whatever you're using, pick a database and upload it once your study is done. Publish your papers, that's great, but also make your data accessible. I think it's a powerful tool that people can use, and having that freely accessible is important. And the last one is, and then I'll stop, one thing that maybe even the pandemic more than anything else has illustrated, even if science knows what we should do, in order to get everyone on board, right, the entire population on board, we need to involve them in decisions, we need to train them, we need to educate them. So that is a big challenge that we're nowadays facing, and I think that's something we all should think about. How can we achieve that we get not just scientific knowledge, but how can we communicate it in a way that people actually base their decisions of it in across the entire population? That's maybe not so easy. With that, I will stop here. If you have a question, I'm happy to answer your question. Thank you so much.
Thank you so much, Dr. Thank you so much, Dr. Schleifer. We have one question from the audience.
Go for it. Yes, I don't see the question. If you could read it, because with the screen sharing, I don't see
it. When you create a defect, can you control their position and shape?
So that's an excellent question. So I think that's at least our hope. So currently, what you saw in this presentation was largely ion beams. I think with ion beams, that goal is a bit difficult. And the reason is, an ion beam, you cannot really particularly aim very well. Now, how is a laser beam different? Because if you use light, you may say, well, a light is even less focused than an ion beam, and maybe you cannot control it at all. Maybe, but there is one effect that we found in those simulations, which is with an ion, with a laser beam, you can excite defects that have specific energy transitions. And if you, so what that means is, by picking your laser frequency, you can excite specific defects. So let's say your sample has, I don't know, a certain type of vacancy present. That vacancy is resonant with a specific excitation energy. And if you use your laser frequency accordingly, you can target those defects. So I don't think that you necessarily can go in and pick one specific defect and move it around, but I think it is doable that you can pick, for instance, a certain group of defects or a certain class of defects by using light with very specific energy characteristics.
Thank you very much, Dr. Andre. Indeed, we are very happy to see you again in the second session of our webinar. Indeed, you are talking about the creation of and manipulation of defect by using the helium ions in order to make an excitation of the electrons. My question here, Dr. Andre, in which limit we can go in this manipulation? If I can more clearer, is there any secondary effect that we cannot monitor during this process, during the excitation process by helium ions?
Yes, so I think when you expose your sample to any ion beam, but also helium ions, one thing that also happens is, if the ions are slow, you get, in addition to the mechanism that I showed you here for energy deposition, you get ion-ion collisions that matter and create defects. The good thing is, I believe in thin materials, that effect is much less important. And the reason is, in a thin material, what really matters is your initial beam energy, because in the thin material, that initial energy will not change so much. And the particular importance, whether electronic deposition of energy or ion-ion scattering is more important, depends very much on the energy. So if you come in with a beam that has the right energy to trigger electronic excitations, that beam will not change its energy so much in a thin material. And so I think for two-dimensional or low-dimensional materials, this mechanism is actually pretty well adapted. If you expose a bulk material to an ion beam, you will eventually slow down these ions enough that they then start to interact with the ions of the target, and that causes a lot of damage.
Okay, thank you very much, Dr. Andre.
Thank you.
Thank you.
Thank you, Dr. Schleifer. And now we will move to our second speaker.
Great. You can take, do I have to stop sharing or can you take over?
Yes, we can take over.
Okay, perfect. Thank you.
So our next speaker is Dr. Siddiqui Noor Al-Ali. He's Associate Professor of Geomorphology, Geography and Geographic Information System Department, College of Art, Imam Abdurrahman bin Faisal University. He is the chairman and session reporter at the GTONS. He is also a member of the Scientific Community of GTONS Conference and a member of the Scientific Committee of an international scientific journal, an active member of the academic societies. His title will be The Effectiveness of Material Recycling in Stabilizing Sand Dunes that Affect the Highways in the Eastern Province.
Dr. Siddiq.
Hello.
Hello. Good morning, doctor. How are you?
Good morning. How are you? Fine.
So now you can share your presentation.
Okay. Okay. Thank you for the weapons and my colleague in these weapons. My name is also, I say that Siddiq Noor Ali, and I say thank you to my colleague, Dr. Badriya Muhammad Umar Al-Habib. And we need to speak about what's happening. Share.
Can you share the presentation, Doctor?
Yeah. Is it clear? Now I share it.
Yeah, the sound is clear, but we need the presentation. Okay.
Yes, I share my presentation. Yeah. Is it clear?
Yes, go ahead, please.
Okay.
Okay.
Okay. We need to speak about the effect of material recycling in the supplies sand dunes that affected or affect highways in the eastern province. We need to speak about theoretical about this topic because he is apart from big studies, and we grasp that as the ferrets, the idea, it is the problem of the sand dune moves. It is a very important and dangerous issues in the eastern province. If you see the eastern province, you see the dunes, it is big problems. And for this, we think that how to solve this problem, because desert, it is big problem as a long regional from Africa to Asia. And if you see that for the climate change, also it is big problem, and how to see the solve. If you think about these problems for the global, you can see it is very danger for the life and for the animal. And so that is many problems from the desert. If you control the desert, you can solve all these dangers. Until to solve, you need some solution. This solution may be things at the local level, and so developer to the regional level, and so that may be covered as the globals. Here, in this case, the desert, it is very important, and how to see, because it is many materials in this desert, and we need to control for this resource without any effect. In Eastern province in Saudi Arabia, the case, it is very especially than the others province, because the Eastern province, it is lay in the location of the Saudi Arabia as Eastern, and for this, you see there is many affected for wind and for tropic. Besides that, negative, because it is a negative affected on development project, specialized as highway lines. This is a side effect. Also, as a pipeline of the petroleum, or a high pipeline also of waters. This is big problems. And if is there any problems, maybe proximate or damage, and how to solve that, you can see many sand and sand dune can be at the end. And the sand creep for Najd desert, or called Sahara al-Najd, or Najd desert, situated towards the eastern province. It must cross the highways, and he lays in the problem. It's causing traffic accident and heavy loss in life insurance. This is one side of these problems. The sand dune also can be problems for the highway. Maybe make a Saudi Arabia, it is if you return back to highway and the accident, you see it is number 2 or 3 as the world. This, it is very cost for the life, which you can be drive the cars, and most of that accident from youth. And this is, it is a resource for Saudi Arabia, which can be depend on to develop it. The other side also, the insurance can be high, because there is a problem for accident for car, and for life also in insurance can be problems. So that the idea, it is about how to take these problems by addressing it is in a scientific paper for various disciplines. Therefore, applied geomorphology, and it is one of the disciplines that intertwine with the environment science and science in feeding a sustainable, smart, scientific solutions. This can be during all about the idea. You see, it is apart from big study, and one of the solution, we think how to resolve this dune without to be dangerous for the driving, and for the animals, for the pipeline, for the pipeline for waters. This we think that we must think how to resolve that. This is the idea. The importance of the study comes from feeding sustainable invited solution to stop the encouragement of the sand in Saudi Arabia as a lost cost. If you think about this source of the wind and the sand, you can return back to see as a global, which is it comes from 2 ways. One, from the north. It is from European air altitude. This is it is back in the northern or in Siberia regional, behind the Tega region, and this is can be push the wind to Saudi Arabia, and unfortunately, it come without any forest or good forest or any big waters can be take for survivors. And the other side, it came from Africa, from Sudanese tropical depressions. It is a low part. We come, it is very hot, and to this it links the north, and from here you can begin to affect it in the airs, can be affected at the most of the geomorphological elements, and for this we can be get it small sand and take with a long period, and we with a long period, it takes that at the enter of the Saudi Arabia. One of these sites we can be rested it, the province, the eastern province. It can be rated at Al-Hasa, and here also and in Al-Qatif. These two ways can be very, very dangerous for the sand dune. The big problems and the process in which this case, the eastern province is the one of the largest province in Saudi Arabia, and the most commercial traffic as the result of the creation of the seaport as the Dammam and Ajibel. Besides that, also there it is airport in Dammam and in Al-Hasa. This is two port. It is very important to be careful with the without to be affected with this sand dune. So the land transport movement is active through the highway, which is exposed now to a wave or creeping sand as a result of the geomorphological and geography of the province on the land. In the end, to study these sands lead to a known and dangerous traffic accident with estimated numbers. According to the general director of the traffic in the province, in the eastern province, we use many approach. First of that, we use GIS. We load it from Landsat from different period from 1985, and the others in 20, and the other in 2060. These three different for period. If you analyze that, you can find the difference we see as the end of the show. We use GIS for the various geographical approach to collect and analysis data. This is for technical, which we can use for it. Besides that, also we use many approach like historical and like to describe these problems. The most important result you can see applying the technical of the using car tools on the sand dune to benefit from them. It is stabilization and planting seeds and disposing of them as waste. with the possibility of adding, trying to give an aesthetic characteristics. It is very important to know about that. If you see here from these figures, the evaluation of the area of the sand dune from 1985 up to 2016. There is one, if you see from left here, this image, you can, if you see and compare between three, this image, it is in 1985. And this second, it is in 20, sorry, in 20. And the last in the 20 and 60. 16, sorry. You see, this, if you analyze this image, you can see the yellow, this is the sand, and the white, you see it is not covered by the sand, and this also, I think it is railway, and this is the highway, and the other, it is second highway or roads. Problems of this case, the sand, you know, as a general, can be moved in Saudi Arabia each in years. I think it is went 15 meters each years. As this means, if you calculate any space and the highway, you see, can be moved early year 15 meters. That means you must be careful to stop this sand. In the other or the second image, if you compare, if you choose this space, if you see here, it is some dunes, it is not be big, but if you see in the 20 image, you see can be white, you see, and if you see the resolution also, the sand be yellow, but here it is high yellow or it is stated the colors. If you see number 3 in 2016, you see it covered, this space can be more than 75%. And if you apply that of all this space in image, you can see there is more advanced and more be covered by the sand, and this is very dangerous. So as this, if you need to resolve that, you must think good for the local. You must think good for at the local level as the first. How to stop this wind. And if you stop the wind, I think that the sand, it can be moved. If you need to resolve that, you must return back to reason of this wind, how to move. And for this, we can talk and we link with tropical and geomorphological and with weathering. We need to resolve and analysis that. And if you analyze that, you can reach at the end what the kind of the reason. Here, unfortunately, Western province it can't covered by any trees or vegetation or forest. It is not good for resolve. And if you think that we need to see the solve for initiative with the many initiative or initiative like the Prince Mohammed bin Salman to agree. I think that it is 100 or 10 milliards trees. How to apply that? We need to think for the all, and we need to choose some places without affected with the sand dune, as this is too difficult. And for that, we think for the good solution, if you see this road or highway, there is many machines in the same of the way, this and this. Early years, in 3 or 4 months, can be worked to move this sand from road to away. And the coming years, the wind can be returned back also and began these machines to move from road to way. This can be circularization by the years. We need to solve that. If we think at the local level, we need to use these roads of the cars. I think Saudi Arabia, it is big country and there is many cars. I think that we need to recycling this ore. I think if you calculate as years, more than 2,000 walls by years, 2,000 walls. This, if you put from this image or this photo, you can see use the wall for agriculture, and here for to sand or like circle, and this I think it is set up to reach this house. That means need these walls to put at the wall after 10 meters, put others walls after 10 meters, put also. This can be covered because other solution, it is traditional, can be used for solution to stop the desertifications. Before we think, and I think that use recycling of petroleum and use the use of the trees and also use the other materials like plastics. Also, it is not good or suitable. If you use wall this of the cars, I think that you can put as 10/8, like 3 or 4 meters, and this is the wind. If he come to these walls, it can be passed. It put in and full it, and it trans the other space. And for this, we need to use this wall to stop the sand dunes. Thank you.
Thank you.
Yes. Thank you. Finish.
Thank you very much, Dr. Satiki. So if anyone has a question, just please type them down in the chat box below. Meanwhile, I do have a question. What about your future studies?
Yes, can hear.
What about your future studies?
Okay. Future studies, we need to develop this study to resolve sand dune. Okay, we need to apply that. I think this is a solution. It is a part of study, but if you need to be sure, we need to apply that in some way, like we choose 2 point J Shia from here. If you travel to Riyadh, I think it's more than 100 kilometers. Or we apply that between airport at Dammam and at Dammam. Also, there is places the sand dunes can be moved and covered the road. We solve that. We need to apply it and to solve the this sand dune. The sand dune, I think it is a big problem for the eastern province. It is a big problem. Can be damage many projects, can be damaged and can be dangerous for the settlement and for the Dammam also. We need to solve that. I think I need to solve that.
Thank you, Doctor. We also have one question from the audience.
Yeah.
Dr. Rida, when you use the tires, the cars to stop the movement of the sand, did you know that tires are carcinogenic materials?
Say again.
Carcinogenic materials.
We use what?
The tires.
Times?
Yes.
Yeah.
The tires that you have them. Do you think that they will pollute the environment?
These tires, if you, yeah, I know that. This all tires, were of car. This can be deposited. It is not good for using. If you need to use this tire, I think that in Saudi Arabia now can be used because useless more than 2 million as years. Now Saudi Arabia can be used just for cement manufacturers. Can be used after recycling in the players and in the Olympic yard. This, I think it is specialized to use. Now we need to use these tires for stop the sand dune, but we know if we need to proximal the effect of these tires, I think that it needs less than, or sorry, more than 150 years to be analysis. For this, it is good and suitable to use this tire to stop. After you stop, also you can recycling again. You can move from sand use to use from other place. For this, it is a life of tires. It is long. It needs more years to analysis.
Okay, thank you so much, Doctor, for your interesting talk. And we will move to the third speaker. Dr. Iman, you have any question?
Thank you.
No, thank you. Go ahead, please.
Okay, so our third speaker today is Dr. Nadia Al-Abdallah. Dr. Nadia is an assistant professor at the College of Science, Imam Abdurrahman bin Faisal University. She's a faculty member at the biology department, and she got her PhD from King Abdulaziz University, Jeddah. She specialized in plant physiology and plant nanotechnology and published more than 20 scientific papers in high-impact journals. Welcome, Dr. Nadia.
Welcome, Dr. Hanan.
Good morning, Dr. Nadia.
Good morning, Dr. Iman. Let me
We are
share the
very happy
screen.
to see you again with us.
Thank you so much. I'll try to share the screen. Okay. Okay. So today, inshallah, we will discuss the possibility of irrigating a plant with seawater and its potential effects. This is our content. By the year 2050, the world population is anticipated to reach more than 10.2 billion, and the most countries are expected to face drought stress and water decline by reaching up to 50% by 2025. Only less than 1% of fresh water is available worldwide. Increasing water leaking is one of the major factors which lead to restricting the plant growth and crop productivity also, and in arid and semi-arid zones such as Saudi Arabia. With the increase in food demand, there is a growing interest in providing sustainable agriculture source. That's why the application of seawater for agricultural crops can be considered as the most realistic alternative or complementary source of irrigation. It is abundantly available water by 96.54% on Earth. Even though seawater contains a high concentration of chloride sodium, reach 75%, which have a negatively influence on the plant, seawater also includes an extensive amount of nutrients that are beneficial and necessary for plant and human also. Dilute seawater when in low levels can be used for irrigation or as a foliar spray also. So our articles aims to explore the possibility of using seawater to enhance plant growth and produce a reasonable yield. Seawater components, it has salinity of nearly 3.5%. Chloride sodium is the most important constituent of seawater. The chloride responds almost 55% of the total salinity of seawater. It reaches 75% of the chloride sodium and 10% of the magnesium chloride. Also, seawater conditions or contains other ions, for example, sulfate, magnesium, calcium, potassium, and carbonate. Seawater irrigation in Saudi Arabia. We know that most land in Saudi Arabia can be classified as arid land. It covers about 2,250,000 square kilometers of the Arabian Peninsula. Agriculture in Saudi Arabia actually has been a great development over the last 3 years. But the shortage of fresh water is one of the main constraints when it comes to developing high crops productivity. So that seawater irrigation is one of the most appropriate management practices for arid areas to meet the increase of food demand. Why irrigation with seawater? We choose irrigate with seawater for many reasons. Irrigation with seawater allows saving a significant amount of fresh water. Also, we have the desalinations, which is the process of removing salt from the seawater, has been excluded as a result of economic conditions, especially in developing countries, because it is expensive process. While in the same while the small scales of seawater irrigation is more suitable and economically available. So seawater can be also excellent alternative water source for irrigation because it is available in a massive amount. Also, even seawater contains a high concentration of chloride. It is rich in most of the elements that plants needed. Also, seawater is considered as a good natural fertilizer, as includes most of the nutrients and microorganisms, live or dead, which have a beneficial effect on crops productions. Are there any positive effects of seawater irrigation? Well, let's see. Moderate seawater treatment resulted in the remarkable accumulation of soluble sugar, amino acid, fatty acids in a crop. The low concentration of seawater enhanced the biofortification, which is a process of increasing the micronutrient of horticulture crops and improve the nutrient situation of the food. Seawater increase the level of secondary metabolites, which encourage defense actions against pathogens and herbivores. Examples for secondary metabolics as pigments, alkaloids, toxins, gums, rubbers, drugs, etc. Also, the secondary metabolites impact light absorptive features for photosynthesis and perceive the cells from injury effects caused by the high energy radiations. The secondary metabolites, which increase by the application of seawater, it is also have importance for the human. It is very useful in the prevention of human diseases like Parkinson, cancer, and other diseases used of the dilute. There is important note we should know about seawater irrigation. The ability of salinity-tolerant plants to cope the seawater stress is strongly improved when the plants are not suddenly shocked, but when we allow to gradually accumulation to salt stress. Here we have some examples of research which they used seawater application on the plants, some crop plants, and it's all the result in different concentration from 5 until it reached 30%, and all this concentration give positive results. It's increased the plant antioxidant substance and secondary metabolics, and also increase the plant growth. Here our experiment, we add 4 concentration of seawater from 0 to 40% and the eggplant seedlings for 30 days. In the end of our experiment, we measure the plant parameters, shoot and root length, fresh and dry weights for shoot and length, and the leaf area. And our results showed that the plants treated with 10% of seawater showed significant increase in the growth parameters compared to the control plants. We can see here in the photo the leaf area. But these parameters decreased with the high levels of seawater from 20 and 40%. So we see the positive effect of adding seawater or using seawater irrigation on the plant, but there are some constraints. Only salt tolerance crops can be grown under seawater conditions. Also, using a pure seawater in a large scale is difficult because of the accumulation of sodium and chloride, which cause toxic influence in majority crops. Also edible products and the deterioration of soil structure. So in conclusion, we can say that seawater irrigation can be beneficial, particularly in arid and semi-arid land. Also, seawater plays an important role in sustaining agricultural production, and it is appropriate and a practical solution facing the global water scarcity challenge. Also, the applications of seawater as a complementary irrigation source in certain concentrations level do not negatively affect the crop quality and productivity. Many conventional crops are salt sensitive, but on the other hand, we can say that some crops can be grown up to 30% seawater concentrations. The seawater can be used as a source of elements for fruit and vegetables productions also. The low levels of seawater are necessary for some plants to reach their optimal yield, and this is an interesting point. So our recommendations: determination of a true salt accumulation in soil need longitudinal studies, because the short-term treatments do not give fully reflect salt accumulation in soil. Also, studies in crop responses to seawater salinity will be necessary to provide more information on plant adaptations. This is our reference, and thank you so much for your listening.
Thank you so much, Dr. Nadia, for this interesting talk. And if anyone has a question, just please type them down in the chat below. Meanwhile, I do have a question, Dr. Nadia.
Yes.
So how many ppt of seawater is appropriate if I want to grow some vegetables and if my farm is kind of high in salinity?
Well, it's depend to the plant, tolerate to the salinity. Some plant can tolerate.
So if I have like 3,000 ppt of salinity in my water, can I grow some vegetables like tomatoes? 3,000 ppt.
For tomatoes?
Yes.
Well, tomatoes, now there's many research that improve that tomato can tolerate the salinity in high levels. So I think it will give you a good result.
Okay, so it's doable.
Yes, yes, of course. Yes, of course, especially for tomato. It's depend to the plant.
Okay, so we have Dr. Rida is asking a question. First, he's saying that take this opportunity to thank the College of 2 days of International Scientific Day. And his question is that when watering plant with seawater, be sure that they will die to salinity. Have you been thinking about it depending on the type of the plant?
Yes, of course. It's need more research about this. It's a new field to use seawaters. It have some risks, so we have to do more research about that.
Okay. Thank you very much, Dr. Nadia.
Okay, thank you so much.
And we will move to speaker number 4. We have Dr. Mikael Sivijärvi. He's the director of ICM Research Institute in Sweden. He has a strong track record in research regarding the growth of advanced materials since 1995. Since 2005, he has gained experience in the entrepreneurial discovery processes by creating startup and technology transfer from research and innovation. He has contributed to establish avenue in Europe research and innovation strategy for smart specialization for general economic growth and strategic national innovation programs. Now he's an active in global platform for business from research, training next generation leaders. Welcome, Dr. Mikael.
Thank you very much. And I'm happy to present here at this conference. Can you hear me?
Yes.
Great. And now I will talk a little bit about the usefulness of research, what we can do to actually achieve the goals in year 2050 and beyond by considering aspects which are related to what we are doing in the research, science and technology today. So I will give a sharing of experiences from my various backgrounds and how I view that we can have a sustainable approach. And I will give examples of approaches which are done internationally today to create impact in the world. And this is, of course, coming from advanced materials and innovation, which is the origin of many things. Materials is the basis for all the technologies. Share my screen. There. So my name is Mikael Syväjärvi, and I come from Sweden. And I have been active in research since 1995, when I was a young PhD student, and then deep into the physics about silicon carbide and graphene and other things we were doing. And like 15, 20 years ago, I started to become interested in making companies or technology transfer from our research. So then I had some experiences in creating startups and the entrepreneurship. And then I also took part in regional development programs and national innovation programs. And I learned a lot, I hope. And by that, I started to consider the broader aspects of research and innovation and making real usefulness from the research. And if we go back in time, then we're coming back to materials, and we have the nitride material with the phosphor, which was developed more than 30 years ago. And that is now the basis for the white LED. And the white LED is, as you know, saving tremendous amounts of energy. So it is a real success story of how materials can be developed to a sustainable technology that is has a global impact. And if we go back more than 30 years, at that time, the researchers did not have any commercial systems. They had to develop their own growth systems in order to be able to develop the materials. So here is a photograph of one of those systems when I was visiting Mayo University. Then they developed the high quality material using a buffer layer and so on. So that was the step to get high quality material. And then at that time, it was very challenging to have the P-type doping. So then they had to develop another machine just for the P-type doping. And that created momentum. And of course, this was very initial research in the beginning. And in the first conferences, there were not many people at all interested in these research findings and so on. But then it grew, it started to have attention. And the blue LED was commercialized in 1993. And then it was developed further and further until we had the white LED, which became commercially available like 10, 15 years ago. And in the beginning, it was not very stable technology, but now it is so mature, so reproducible, and you have the high volume production that this is available in the society to save tremendous amounts of energy. And the background there resulted in the Nobel Prize in Physics in 2014 for these things. And most interesting, this technology now is used also in other avenues. For example, art and paintings. So when we have the LED, the light is directed towards paintings and so on. We can have different experiences of these paintings just by changing the light, the frequency, the wavelength, and the direction, and so on. So this becomes another avenue in design, not only energy saving. And also for the city development, now we can have LEDs all over the place. They don't consume so much energy. And here is an example from a building in Brussels. It changes light every 3 seconds or so. So it is used also for the smart cities experiences and design and all those things. So the technology broadens very, very much. And what is happening now in Japan, I heard a talk by Professor Amano, one of the Nobel Prize winners, at European MRS spring meeting in 2019. Then they were talking about how to come to the next level. So then the investors and funding agencies, they wanted more of these key enabling technologies, which are they have been developing from the white LED. But they don't want it that it takes 30 years. They want it in less than 10 years. And Professor Amano said that, oh no, this is not possible. It takes a lot of time to develop material, device, and go to implementation. So what started then was that there are programs where students are encouraged to be entrepreneurs if they have that interest. And those students who would like to reproduce the research findings to production, those are encouraged. And those students who want to invent new technologies, they are encouraged. And to this program, you have internships, mentors, and there's a lot of things. And this is one of the sustainable approaches to already now encourage at early stage to make usefulness from the research and use the young people. And if we are targeting the goals of 2050, we will definitely need the young people. 2050, I will be more than 80 years old. So then my role is more to contribute now what can be done. And another example where the research to business is already included is in the graphene. And in Europe, there's a graphene flagship program. So European Commission has put 1 billion euro for graphene to go from research to market. And there is a plan for this, so a 10-year plan, first from the material, then to device, and then to system. And after 10 years, it should be self-sustainable. It should be moving on its own terms. No more needing a funding from European Commission and so on. So that is the plan already at this stage. And in the beginning, if you remember, graphene was so super exciting and there were revolutionary findings more or less every day 10 years ago. But that was in research. And still we don't have so many products which we can buy. And this is a typical example of what we had to think of when we have emerging technologies from advanced materials. In the beginning, there is research findings, and that is exciting. But it is not in production, and there are no products. And then during this time of making reproducible research findings, and then go to production and think of volume, then not much happens. So this is the famous valley of death, which takes a few years. And then over time, it will increase its maturity and go to higher technology readiness levels. And this is what we are experiencing today, and it takes many years. We have to face the truth about that. It's a fact that it takes many years. And my own example, where I have experienced this from the beginning, was silicon carbide. In the 90s, it was a very hot topic, lots of research, not many things were done. I was involved in the growth of the material, so everything we did became a study and we could publish papers and so on. So it was a very hot research topic. And in the 90s, there were no companies from the beginning. Some companies were interested, then some companies started to be formed and so on. And then now, a few years, a large number of years later, it is a market. And the idea at that time was to make available wafers, and then we have wafers available for epitaxy, and then you make devices. And my research was about using the general growth method. I will not go into the detail, but instead of growing bulk material to produce wafers, we use the same concept, PVT concept, to make thick layers. And at that time, it was interest to have thick layers for the power devices. And power devices is the market of silicon carbide today. But that method, which was my PhD thesis in 1999, was not successful because we had thick layers, good quality, but it was not pure enough. But now, a few years later, we are using this approach to make fluorescent silicon carbide, light-emitting silicon carbide. And this is done by adding elements into the material. And here's another example which I would like to share, because that research which I initiated in Europe in 2010 as a research collaboration, and I coordinated the network there and became a partnership. We have collaborations, nice collaborations, and then we continue to apply for projects together. So this is a consortium which is still active to produce new ideas. So here you have Hayano, who's the coordinator of one of the European projects now. We worked together, and then she proposed to use silicon carbide for frequency comb. This is just an example that from the original ideas and collaborations, we can come to new avenues. And frequency comb is a device which takes a laser light, splits it to several wavelengths. In that way, you can have much more data traffic, more communication of data, and you can reach much, much higher data traffic, up to 1000 terabit per second, if you're using the frequency comb. So it's a revolutionary concept which hopefully will be implemented. And if it will not be implemented, we will learn a lot. So this is a European research project. And it comes from the material of silicon carbide. So once we have established consortium, we go to new avenues, which will create a cascade of research findings. And in our case, we are doing the growth of silicon carbide. We are using high temperature processing. And in that one, we have a wafer of silicon carbide. We heat it up, silicon and carbon leaves the surface, and more silicon than carbon leaves the surface. And then we have one layer of carbon on the surface of silicon carbide wafer. This is graphene. And the first time we had graphene was 2005. And since then, the group has explored various avenues. And just to give an example of what is in the future of these things, is that once we modify the graphene on silicon carbide, for example, by ion irradiation, we can tailor it for the different sensing purposes. So this is a research area which we would like to continue to go further to. And if we are thinking ahead, then we can also have other carbon materials, integrate them on the graphene on silicon carbide, and make a platform from which many new things possibly could be developed. And for this, we need the young researchers who will have the new ideas. And the second thing which we have to think of as a researcher today is that we are creating research towards technology. We describe a value chain and more or less hope that this will be used in the society. And now we should motivate more how our research fits to the society. And in Europe, there is a regional development program, the Smart Specialization. So this is for regional development. But when regions find their strengths, they find actors. The actors are making value chain together. And that is the way to develop the region when there's more business from the value chain. So the research is describing a value chain from its technical purpose, and the region is describing from the regional relevance. But there's no communication here that we can show that, oh, both are active. In that way, we can create synergies and motivations for the future by considering the smart specialization for regional development and the technology and research. So in such way, there are similar descriptions from different points of view. And if we are emphasizing that both are doing similar things, same things, we want the same things, there will be more impact by joining the forces. And this impact will have such direction that we can actually motivate that here the technology fits in a regional context, and when it fits in the regional context, then it fits in a bigger context, and so on, that we actually can show that here's a line where we provide a possible contribution of our technology to the Agenda 2030 goals. So it's a more clear motivation instead of just taking one of the goals and saying that, yes, we will make more sustainable cities. If someone asks, how will you make a more sustainable city? What is your contribution? If we motivate it by the regional relevance and those things, then we can say that our technology fits in the regional context. The regional context has a city context, national context, and innovation context. So this chain is important to be aware of. As researchers, we don't need to know all the details and so on, but we should be aware that they are and have the continuous discussion with the policymakers, citizens, and so on. So this exchange is very, very important. And in my case, I became personally interested in the business from research, how to make entrepreneurial research companies and some usefulness. So I don't have the same interest to publish papers like before. And I think that the publishing of papers, this will also change. For example, when we aim at publishing in high impact factor journal, this takes a lot of time. And after 1 year, 2 years or so, it will be published. But there will be no one calling from the other side of the world to offer us a business. It will be offered by someone who is in the local context, in my region or in my country. That's how the business from research will come. So then it can be more efficient to actually make it visible regionally and nationally that we have these research findings, and that can have more value than high impact factor journals. So this is something which will change over time, together with the open publication, open innovation, open information, and more openness overall. And if we can have contributions from the young people about these things, for example, young person A here on the left side, then that is a PhD student who's motivated. And then they continue, they collaborate, they have interaction, they become a little bit more senior. Maybe they will start to have a PhD student, maybe they will have a postdoc, and then those continue, collaborate, create impact, and then the PhD student of the PhD student will have a PhD student or postdoc. So then there's a cascade of effect. And this we can only create if we're encouraging the young people to do the research. We give them lead. And me, I'm probably too old to be contributing on the right side here. So the best thing I can do is to support the young people so they will get momentum as fast as possible and get going. So this is a sustainable approach for 2030 and 2050 that we have to think in a little bit different ways. But again, it will be that we have to have something which is a finding. And when we go back, this will come from the research and science. So the things we are doing today, they will have impact. And especially when we meet people from other disciplines and when we get new ideas about things, then new avenues will emerge in unexpected ways. So this, what we are doing today, and exchanging information like at this conference, this is the basic for the new contributions to the year 2050, 2030, and so on. So with this, I would like to thanks for this opportunity to present. And I hope that I have given you a flavor that we have to think in a little bit new ways about what we are doing, and then understanding the various ecosystems so we can learn from each other and so on. So and in that way, there will be more impact from the research and innovation and advanced materials in the future. So thank you very much.
Thank you very much, Dr. Mikhail. So I have, I think, two questions. Here, Dr. Mikhail, you are talking about encouraging young people to join the research field. So as a director of a research center, so what are your strategies to encourage those young research to continue in the new field mainly? This is from one side. From the other side, what are the hot topics that you are planning to tackle in the next few years in your research center?
Yes, the main thing about how to encourage young people is, of course, always to be positive and give them opportunities. But also, sometimes the young people are not thinking that they can be active to create momentum. And one example which I give is that when there are conferences and symposiums, like we are organizing such, then one can propose symposium, and it doesn't need to be a professor who proposes a topic and gathers people for the symposium.
So by giving them the initiative.
Yes, so if we support them, take the initiative, and we will help you to find and so on, then they will be coordinating the symposium session, which means that people will come and ask them things, and they will be increasing their network. So then when we share our network that here is a nice symposium, maybe you should go there, then the young people can have more network. And when you have more network and people come and meet and discuss, then there will be ideas for collaborations and so on. So in this way, to go away a little bit from the professor type of thinking that always, not always, but the professor is the most, the one who attracts people. And this is fine because it's a name, but usually they don't have time. So we should allow in different ways the young people. And now we are also starting with IAM, we're starting a support program for young people to be trained. Because the usual case is that there are nice research ideas, but they're specific. And if we are then sharing our experiences, like I'm doing here today, then they will think about the research topic in a larger context. And this is very, very important when you apply for projects, because the most important part in a proposal is the impact. How will this research make a difference? So if we can motivate this in a better way, then it is a higher probability that the proposal will be accepted. So such things we can basically sharing experiences and knowledges to each other in a more open way. So this is the main way. And for the future hot topics, I think there are the combinations of materials, and the more we go towards nano, the more things we can do to actually contribute. Usually you have the white LED. This is a big energy saving part, but it is not enough to save the world, because then we need additional complementary energy saving technologies. And you have the big energy saving, you have the medium energy saving, and we will need the small energy savings. So when you have all these together, then we will be more sustainable with the future. And also to create new research avenues from the local context. Like I'm living on the countryside, so out here there's nature, and then we have a heat plant, and we are using forest to burn it to make heat and heat the houses. Then we can study the process of heating and how the ashes from this heating, burning, can be used for the ecosystem and the rural development. maybe the ashes can be used for some new nano devices because of their specific nature, and so on.
Thank you very much, Dr. Mikhail. Indeed, I have just a last question here. How you can succeed the business from the research, of course, as a director of research center? And if you can give us such examples.
Yes, what we do when we have an idea about research and then a finding, and then we believe that this could be a possible business case. So then the main thing is to get started and then move in this direction. And in many cases, the actual business from the research, it's another thing which happens along the way. So it depends on how does the global situation look like, the politics, and who wants to pay for something which you have. And the only way to find this out is to start moving, doing something, and trying to make business. But I have several examples which I have seen from colleagues and so on. When you start activity, then it's a small part of the things we're doing which someone would like to pay for. And then it became a market for something totally else than we were thinking from the beginning. But we would never find this out if we don't start moving. So this is the only thing I can say, because there's no guarantee or recipe or anything how to make business from the research. So start to move, and also to network and collaborate, because then there will be new ideas. Someone will propose and ask, why don't you try these things in this way? And then maybe that will be the opportunity. So this networking and sharing discussions like here at conferences is so super important.
Excellent, perfect. We have a question from Dr. Rida. He said for PhD student, published paper is more important than thinking about applied science and doing research business. So he asked how you can convince them, Dr. Mikhail.
We will still need to have both activities to publish papers and so on. And something which we can do more of is actually to share the learnings from the paper, for example, to the society. So if we have a nice paper, many people in the society will not understand what this means.
Yes.
But if we explain it to them in a simplified way, then someone will be interested. Like when I have the fluorescent silicon carbide, then I talk about the light emitting silicon carbide. We add some elements, and then we get light out of silicon carbide, and it's a new type of light with the pure properties. So it's a pure white light. And such things, people outside research, then they will understand. So this being active outside the research channels, this should be done in parallel. Then there will be opportunities. Then you can still publish, but you do more work to actually about the dissemination of the findings and write it in simplified ways. And even small things, when you're on LinkedIn, then you can make a post about something, and this will raise awareness. And in some cases, when you were not planning to write, but you wrote something just in the moment, that will be spreading more than the planned dissemination. So this is same thing, but we do a little bit extra regarding the visibility of the research findings. But still, we need the papers and so on, because this is the core of our expertise. When the business people or media or citizens or policymakers comes and ask us, then we have to have our expertise. And this is the publishing of papers. So it's still needed, but we need to do additional dissemination.
Thank you very much, Dr. Mikhail. Please, Dr. Hanan, go ahead.
Dr. Mikhail, there is also the idea of publishing a scientific paper in a journal and then maybe writing down the same idea in more simple language for the local newspapers, for the community to understand what kind of research is being done. What do you think about that?
This is excellent, because that is what I was just talking about, that we need to simplify this. And actually, one thing which is now growing is presentations of video. So if we make a presentation, and if we know that this is not for our research colleagues who are experts, then we will simplify it. But in several cases, it's easier to get the idea when someone is talking. For example, when I learn something about have interest in making music and how to mix and master and these things, I often go to YouTube. And then someone is talking and showing. So then that lowers the barrier of how to understand things. And today, it is very easy to make a video. We can make it ourselves and upload on YouTube and so on, because we have the mobile phones. So it's not so difficult to make it. And it doesn't need to be perfect, like a planned professional video. It is sufficient to make something simplified. And I made, for example, one when I was in my office. Then I had a crystal, which I found in my drawer, which was grown maybe in 1996. So then I made a simplified video. Here's a silicon carbide crystal. We grew it many years ago. It has a lot of defects and so on. 1.5 minute video. But this is the video which has most views in my YouTube channel, even though it was just made in a moment and uploaded. And other ones which are planned more, they don't have as many views, so on. And also there is
I agree with you.
Publishing of video presentations like we are having, that it is published and indexed, and then you have the publishing credits for it also as video. So this is also coming. So such things one can try and become more familiarized with. In the beginning, it's very scary to make a video where you talk about things and release it to the world. But once we have done it a few times, then we will be more experienced. And then we also know that, oh, the other people, they're also humans. I'm a human, so it's fine if I say some mistake and so on. But then we are sharing in an open, honest way, in a personalized way.
Yes, I agree with you. And it's also nice to make these small documentary videos in the labs, not only for the community, but also for the young researchers who does not have the expertise.
Yes, and also it's a nice memory. And I'm so amazed when you have these documentaries about people, like 30 years from now, there will be a documentary about some of us. I don't understand, how have they found the videos from where they were kids, small, from the lab? Then they must have planned these things already, so that they took videos and things, so they could make a documentary of the findings. But we cannot start with those things 30 years from now. We have to do it today. So using the mobile phones, and it's okay that it's shaky and moving, but it will be a part of those documentaries in the future. So it will be useful more than we think.
Thank you very much, Dr. Mikhail Sivijärvi, for your interesting talk and for joining us today.
Thank you.
Now we will move to the next speaker. We have Dr. Mohammed Ouais. Dr. Fakir. Then we will move to Dr. Fakir Mohammed. Are you with us?
Yes.
Yes. Okay. So he has more than 25 years of teaching and research experience in different national and international organizations. To date, Dr. Mohammed has 125 research articles, 3 patents, and 8 books. He is the main research interest include developing of polymer nanoparticle for drug discovery, and he is a recipient of various national and international awards. Today, he will speak to us about the use of nanoparticles system for biodetoxification. Thank you for joining us, Dr. Fakir. If you can please share the presentation.
Thank you very much. Okay. Right, let my presentation. I'm trying to share. I don't know, it's
It's okay. Take your time, doctor.
mute. Okay, yeah. I think
The sound is breaking a
there
little bit.
maybe I'll
Dr. Mohammed, I think that the sound is interrupting there.
okay. Are you listening me?
Yes.
Okay, but I'm trying to share my presentation, but still.
Okay. Take your time, doctor.
It's, I don't know how long it will take.
If you can download the page again.
Also, maybe because of the video, if you can stop the video, it will share faster. Yeah.
Yeah, that's fine. And then okay. Okay. Can you see my presentation now?
Yes, doctor. We see the presentation.
Okay. All right. All right. So, so should I start now?
Yes, please.
Okay. Thank you. All right. So, this is Dr. Fakir Muhammad from Bahauddin Zikra University, Multan, Pakistan. I want to thank you, the organizers of this webinar, for providing me a chance to speak today this conference. The topic of today's talk is nanoparticulate systems that are used in the biodetoxification. So we are exposed to a number of toxic substances in our daily life. And according to the United States National Poison Data System, around 2.5 million cases were reported into the system, including 2.13 million human exposures and around 57,000 animal exposures. Amongst the major classes of toxic substances that are usually reported into the system includes analgesics, 10.3%, household cleaning substances, 8.4%, cosmetics, personal care products, 6.5%, antipsychotics, sedatives or hypnotic drugs, 4.9%, antidepressants, 5.3%, and pesticides, 3.6%. So these are some of the major classes of toxic substances that can produce toxicities. And different strategies, the old strategies such as using the gastric lavage or using some non-specific antidotal therapies are outdated. And for the last 2 decades, some nanoparticulate systems are getting importance to be used in biodetoxification. So we can classify these systems into 4 major categories, including lipid crystalline nanoassemblies, liposomes, micellar nanocarriers, and ligand-based nanocarrier systems. And we can divide these crystalline assemblies into hexosomes, spongosomes, cubosomes, isosomes. And the micellar nanocarriers can further be subdivided into lipid nanocapsules and microemulsions, while the ligand-based nanocarrier systems includes nanoreactors, nanoconjugates, nanobodies, nanochelators, nanospheres, nanocomposites, and nanosponges. So I will share briefly with some of these nanotechniques that are used to detoxify the biosystems, and then at the end I will share some of my personal research work in this context. The mesoporous silica nanoparticles, these can detoxify the toxic substances by advantage of the metabolic reactions, including phase 1 and phase 2 conjugation reactions, where they can detoxify the toxic substances by oxidation reduction or metabolic reactions or some other glutathione conjugation or glucuronic acid conjugation. The mesoporous silica nanotubes are extensively used to detoxify the toxic substances, and once the toxic substances is engulfed into these tubes, the detoxify reaction takes place. And after the detoxification, the substances are diffused out of these nanotubes. In this way, they can be used to detoxify the biosystems. Another strategy that is used for the detoxification is the nanotubes. We know that these are the nano antibodies. These nano antibodies have certain specific regions that can bind certain venoms or toxins, such as scorpion toxin or snake venoms, and they can bind like antigen antibody reaction, and they can neutralize the toxins in the bodies. Similarly, another technique that is called as conjugation thiol modified DNA, where gold nanoparticles has the ability to conjugate certain other anti-cancerous drugs and can detoxify the biosystems. Excuse me. Then there are certain biomimetic nanoparticulate systems, including nanocomposites. And these nanocomposites, they are specifically three-dimensional synthesized nanostructures, and they have certain liver hexacubal structure, and specifically they can entangle many types of toxic substances and can be successfully used to detoxify various biosystems. From University of California, San Diego, where they use this innovative technique by wrapping the polymer nanoparticles with biomimetic membranes, and then they have used this nanosponge in order to detoxify certain bacterial toxins from biosystems. So in spite of this technology, we used in our lab to see that if we can use this nanosponge or nanoabsorbent for cellular detoxification of chemotherapeutic agents. As we know that the chemotherapy has been widely accepted and used as a clinical treatment for tumor management. Apart from cancer cells, the large majority of chemotherapeutic agents can also penetrate into the healthy cells, resulting in adverse side effects. So nearly 40% of the toxic exposures in humans opportunities available in the clinics. So the nanoparticle-based approaches are suitable alternatives. So we try to use this nanosponge system in order to detoxify anti-cancerous drugs such as doxorubicin. We first of all prepared this red blood cell membrane. We isolated the red blood cell membranes, and then we used the PLGA, polylactic-glycolic acid. This is a polymer, and this is the only polymer that is biodegradable and biocompatible and is approved by US FDA for in vivo administrations. So we coated the cell membranes onto the surface of these polymeric nanoparticles through extrusion process, and then we used this nano construct. We named it as nano absorbent in order to study the detoxification of chemotherapy. The figure B is the B part of this figure indicates the size of these nanoparticles, that is around 140 nanometer, and the C part indicates the transmission electron micrograph of these membrane-coated nanoparticles. Here we can see clearly the PLGA core wrapped with this biomembranes. And in the D part of this figure is stability studies of this nano construct, where we see that the PLGA nanoparticles alone are highly unstable in the serum, while the nano absorbent, after coating with these biological membranes, this is pretty much stable in the biosystem. Then we measured the zeta potential, the charge on these nanoparticles, and we found that these PLGA nanoparticles are negatively charged. The charge on this is -50, and the red blood cell membranes are also negatively charged. It's the charge of these membranes is around -20, and the blue line in this graph indicates the RBC's membrane-coated nanoparticles. So this is in between these red and green, so indicating that the size is in between these two demarcations, is indicating that successful coating of the red blood cell membranes onto the surface of the PLGA cores. So in order to see whether this nanoconstruct has successfully retained the major proteins of biological membranes onto the surface of these membranes, we run this STS page, and here you can see these are the major proteins of the nano-absorbent, and this line indicates the major proteins of the pure red blood cells, and you can see the major bands are there on the nano-absorbent, indicates our nano formulations has the capacity to retain these major proteins of the biological membranes or the cell membranes. Then we studied the absorption potential of a nano formulation against two chemotherapeutic drugs. One is doxorubicin and other is the methotrexate. We incubated the nano formulations, different concentrations with this nanoparticles and drugs. And this graph, upper graph, indicates that PLGA nanoformulation was able to absorb about 80% of this doxorubicin drug, while the absorption of other drug, methotrexate, is less than 20%. So this makes sense that since doxorubicin is a cationic drug, and the nanoformulation is negatively charged, so this is the electrostatic attraction is the main driving force for the absorption of the doxorubicin. So we further studied the doxorubicin with our nanoformulation and not with the methotrexate, because methotrexate is inactively charged, so electrostatic repulsion is the main force for not absorbing the methotrexate by this nanoformulation. Figure C indicates where we studied that whether this nanoformulation is able to absorb the doxorubicin drug onto the surface of the nanoformulation, or it can attract the drug deep into the core. And this zeta size potential measurements indicates that there is significant shift in the zeta potential from -46 to think that doxorubicin has the potential to be sunken deep into the of the nanoparticles. This option can go in or not. Then we used melanoma cells, melanoma cancers, the line used particularly as a line, and we incubated. We actually stained fluorescent. We use different fluorescent dyes such as DAPI, rhodamine, red, different fluorescent dyes, and we stained the membranes and core by Z, then with these cells with the Z-stacking merging indicates that perinuclear co-localization of the nanoconstruct in the cell cultures. And not only this, the Z-stacking indicates that our nanoconstruct has the ability to go inside the cells and has the potential to absorb the doxorubicin drug from the cell cultures. This is the cell viability studies, and you can see that our nanoformulation resulted in the survival of these melanoma cells when we incubated these melanoma cells with different formulations. So after studying the drug absorption potential of our nanoformulation, we studied this potential against certain pesticides that this formulation can also be used to an organ phosphate
Dr. Muhammad, Dr. Fakir Muhammad.
Pesticide drug, and we perform. Is there any time constraint?
No. Just the voice is interrupting. Your voice.
Voice. Okay. Should I speak loudly? Are you listening me now?
No, it's okay. Yes, now it's okay.
Okay. All right. So I try to speak close to the microphone. All right.
Perfect.
Okay.
Okay.
So I'm trying to wrap up my studies. All right. So here we used this nano formulation in order to detoxify certain pesticides, such as chlorpyrifos, and we studied this survival of. We use our experimental animal rabbits, where we administered chlorpyrifos to the rabbits, the lethal dose. And then we used our nano formulation along with these chlorpyrifos, and this survival curve indicates that our nano formulation has the ability to protect 100% of the rabbits from this pesticide. And this the mechanism behind this protection is that the cell membranes has acetylcholine receptors, acetylcholine esterase enzyme receptors, and that can bind the chlorpyrifos, and then the toxic reactions that are usually due to the inhibition of this enzyme, due to the accumulation of acetylcholine, and this nanoconstruct can bind this chlorpyrifos pesticide and can revive can degrade the acetylcholine, and thus can protect the animals from this toxic substance. Similarly, in our lab, we tried another type of nanoparticle system that is called as chitosan nanoparticle population paper, and here these are the kidneys, and you can see that there is significant protection of the formulation. We also used certain drug residues. Once the drugs are antibiotics, these are used in animals, food animals, and their milk products are edible tissues. They can contain the objectionable drug residues, and in this way they can render the food unfit for human use. And this is the one of the bigger barrier in the export of products, milk, meat, and eggs. This is the regulatory requirement, and the sophisticated techniques that are used to measure the drug residues, such as HPLC, are expensive and time-consuming. So we got a detection kit with the use of gold nanoparticles. This is a lateral flow assay-based kit where we can detect 5 antibiotics residues in the milk, such as sulfonamides, penicillins, cephalosporins, aminoglycosides, and I have filed patent for these, and in this way, it's used in the field to see whether our milk is contaminated with the drugs or not. Okay, so these were the funding agencies who supported my research work: the Pakistan Higher Education Commission, Pakistan Agriculture Research Council, and Pakistan Science Foundation, who supported my research work in the lab. And this is all that I want to say. Thank you so much. And if you have any questions, you are most welcome.
Thank you very much, Dr. Fakir. And if anyone has a question, just please type them in the chat box below. Meanwhile, Dr. Iman, you have any question?
Indeed, I congratulate Dr. Mohammed Fakir for this interesting presentation, also for this important record of publication. I congratulate him again. Indeed, I do not have a question for the moment, but I can pass the floor for Dr. Rida, I think. I think that there's a question in the box.
Actually, no. Actually, I did not ask any question because the sound is stopping sometimes to times, so I did not hear it well.
Okay. Okay, welcome.
Assalamu alaikum, Dr. Fakir. I have one question, please.
Yes.
You are using these drugs. That how can we save maximum healthy cells, and how can we damage maximum cancer cell by using these drugs? Can you hear me, Professor Fakir Muhammad?
Yes, we hear you, Dr. Gulbahar.
Yes.
These chemotherapy.
Okay, your sound is lagging. We can't hear you clearly, Professor Fakir. So it's okay.
Yes. Okay. Also important. You're not listening me?
Yes, please.
Now we can hear you.
Now we hear you.
Okay. All right. So I want to say that this very important question that Professor asked me. Actually, the chemotherapeutic agents, the anti-cancer drugs, these are used in the cancerous patients, such as doxorubicin or methotrexate. These are normally used in the chemotherapies. What is the problem that once these drugs are used in these patients, no doubt they have greater affinity for the cancerous cells. They destroy the cancerous cells, but they also have the potential to destroy the healthy tissues, healthy cells. And you have seen that all those cancerous patients who are going through the chemotherapies, their life sometimes become very miserable because of the toxic effects of these drugs on the healthy tissues. So efforts are being tried through this nanotechnology or nanoformulations that if we can use such types of formulations along with these chemotherapies, the toxic effects to the healthy tissues might be, might be minimal, or we can improve the chemotherapy of these patients. This is the main objective of such studies. As I mentioned, that about 40% of the drugs they can produce toxic effects in the healthy tissues. These are the reports available so far.
Thank you very much, Professor Fakir, for your answer. Thank you.
Okay, thank you.
Thank you very much. And now we will move to the last speaker of the first session in the first day. And I would ask everyone, please, again, at the end of the session, we will paste a small link for sending the attendees the certificate of attending the webinar. So in each session, we will send one certificate. Now we have Dr. Mohammed Ouais. He is an associate professor of pathobiology and director of experimental livestock farm at the Faculty of Multan, Pakistan. His research focused on the epidermal biological mapping and control of zoonotic diseases in economically divided communities in the country. To date, he has published more than 60 research articles in international reputed journals, and he's also the recipient of various international and national awards. Today, he will speak about the essential zones, the threats of global food safety. We welcome you, Dr. Mohammed.
Yeah, I can listen to you. So I'm just sharing my presentation.
Thank you.
It's shared now.
Yes.
Can you see my presentation?
Yes.
Yes.
Go ahead, please.
Okay. I'm Dr. Mia Mohammed Awais from faculty. Can you listen to me?
Yes, Dr. Mohammed, please go ahead. Just if you can, Dr. Mohammed, make a full share, full screen. From the PowerPoint. Full screen share.
Can you see it now?
From the icon, yes, yes, perfect.
Okay. Okay, Assalamu alaikum. I'm Dr. Mia Mohammed Awais, currently working as Associate Professor and in charge One Health Research Lab at Department of Pathobiology, Faculty of Veterinary Sciences, Bahauddin Zakari University, Multan. And these are some of my other affiliations. So first of all, moving towards my presentation, I would like to thank the organizers of this webinar for providing me this opportunity to talk on this important issue of foodborne parasitic zoonosis that is a global threat to food safety. So first of all, I would like to share what constitutes basically zoonosis. Zoonosis is a phenomena which refers to the transmission of diseases across the species, either from animals to human beings or from human beings to animals. Except when it is from animals to human beings, we call it exclusively as zoonosis, but when it is from human beings to animals, we call it as reverse zoonosis or anthroponosis. So as we'll talk on parasitic zoonosis, so it refers to the transmission of parasitic diseases across the species in the same manner which I have explained earlier. And as far as foodborne parasitic zoonosis are concerned, these refer to the transmission of parasitic diseases across the species by ingestion of contaminated food and water commodities. So despite of advancements in the hygiene, consumer knowledge, food treatment, processing, foodborne, even then foodborne diseases mediated by pathogenic microorganisms or microbial toxins still represent a significant threat to public health worldwide. So they are causing the enormous impact on the health and livelihood of people around the globe, particularly in the developing and underdeveloped countries of the world. So we have a great concern in this regard with respect to consumers, producers, and policymakers. So in this regard, if we talk about the foodborne zoonotic illnesses, the most risky food chains are animal source of food and fresh vegetables, which are contaminated with human or animal waste, and enteric parasites are the major contributors in this regard. So as literature shows, 60% of known human diseases originate from animals, and 75% means every 3 out of 4 diseases of recent emerging infectious diseases are zoonotic in nature, means they are transmitted from animals to human beings. And 80% of agents with and 80% of such agents have potential bioterrorist activities which are used and are being used as a part of Cold War throughout the world. So what are the economic significance of foodborne parasitic zoonosis? According to Center for Disease Control and Prevention, United States has 28,000. So most of the time we'll have to take the data from other countries which is available in the form of published form to highlight or to show the importance of these issues. Well, as I have shown the data of United States, but unfortunately we lack similar data at global level, that is throughout the world. Anyhow, a rough exploration suggests that there could be at least 1 billion such illnesses worldwide each year. So this is the data regarding the etiologies of waterborne zoonotic diseases in United States. So this first pie graph is showing data from 1971 to 1990. Shows that 18% foodborne illnesses are due to giardiasis, that is caused by Giardia lamblia, a protozoal parasite, and the chemical poisoning contributes 10%, other diseases 7%, shigellosis 6%, hepatitis A it contributes 4%. Then the norovirus, it contributes 3%, and unidentified etiologies, these contributes 52%. And the data from 1991 to 2002 reveals that the giardiasis contributes 12%, and cryptosporidiosis, that is caused by Cryptosporidium parvum, it contributes 7%. So my main focus will be on the parasitic zoonosis. So it as a collectively, it shows that 19% of foodborne illnesses are due to these protozoan parasites, which are Giardia and the Cryptosporidium. So what is the significance? One of the major causes, the foodborne parasitic zoonosis is one of the major causes of mortality, especially in developing countries, as I mentioned. They cause economic losses in terms of loss of productivity and absenteeism. Like data obtained from a study conducted in Thailand, which revealed that wages lost due to high frequency of opisthochiasis, that is caused by Opisthochus species. It is 65 million US per annum, and the cost incurred on medical care is whereas hospitalization cost is concerned variable upon healthcare services required in different ailments or infections. Then these zoonotic infections also cause losses in the agriculture sector due to effect on animal health. So the acute fascioliasis, that is an important cause of morbidity and mortality in cattle. It leads to less productivity and weight gain in cattle. And a study in Peru, it revealed 11 million US dollars losses per annum. So also they cause potential losses in food, particularly the fish and meat export market, which result in the form of ban on the export of food products from endemic areas. And usually such bans have been imposed in the developing and underdeveloped countries due to more frequent zoonotic infections in their animal populations. So this is the incidence of major foodborne parasitic zoonosis
Dr. Owais?
Listen to me?
The sound is lagging, Dr.
listening me?
Owais. Now it's okay.
No, it's okay. Okay.
Maybe without the video,
So
doctor, we will have a better sound.
This is a brief event.
Dr. Owais, please, can you turn off your video so we can hear you?
Yes.
Okay.
I'm unable to.
Just make a stop video so we can hear you in a better way.
Video, doctor. Stop video.
Okay, just a minute.
Stop video icon. Yes.
I'm not getting my video icon over here.
Just down. There's okay, perfect. Okay. Thank you very much.
Okay. So now my presentation is in slide share.
Yes. Just make a full screen share.
Okay. Is it okay now?
Yes, perfect.
Okay. This I have developed a brief inventory of zoonotic parasites which are transferred in different foods. So as we can classify the parasites into different classes like protozoan parasites, nematodes, trematodes, and cestodes. Accordingly, I have developed this inventory or list of the parasites like Toxoplasma gondii. It can be transmitted through water, meat, milk, fruit, vegetables, and shellfish. Then the Cryptosporidium parvum, it can also be transmitted through same food commodities than the Giardia lamblia and Entamoeba histolytica, which are gut infection. They are transmitted by ingestion of contaminated fruit, vegetables, water, and shellfish. Then Blantidium coli and Cyclospora species, these are also transmitted in human population through contaminated water, fruits, and vegetables.
Dr. Uwais? Dr. Uwais, there's no sound from your part.
Hello, Dr. Saab, we can't hear you, Dr. Saab. Hello, Dr. Uwais.
Then the last category is for the... Can you listen to me?
Please check your... Now
yes.
We can hear you. Yes, now we can listen. Yes, please.
Okay, the next category is for the cestodes, in which there are Taenia species, Echinococcus species, and Diphyllobothrium, which are transmitted by ingestion of undercooked or raw beef. For example, Taenia saginata is transmitted by ingestion of undercooked or raw beef, and infection is transmitted by ingestion of raw pork meat. And the Echinococcus species and Diphyllobothrium species are transmitted by ingestion of raw fruit and squids. So as far as the water commodities are concerned, so the contaminated water can lead to several GIT infections like cryptosporidiosis. So this giardiasis is caused by Giardia lamblia, that is also known as Giardia intestinalis or duodenalis. It is transmitted by intake or ingestion of contaminated water and food commodities. It is also known as a disease of poverty or traveler's diarrhea, because the people from industrialized or urban parts of the world, when they travel to the endemic areas, they get infected by ingestion of the contaminated water. So that is why it is traveler diarrhea. 7 days pre-patent periods, it caused GIT disturbance like diarrhea in 60 to 90% cases, then abdominal pain, cramping, bloating, muscle pain, fatigue, nausea that might accompanied with the vomiting or maybe not. Then fever might be an unusual sign. Then the most important is the foul smelling stool of the affected individuals. Then loss of weight and stunted growth in the children. Then the next one is the cryptosporidiosis, that is caused by Cryptosporidium parvum. Earlier, it was considered that Cryptosporidium has only one species, that is Cryptosporidium parvum, but with the advancements in the genetic characterization of the pathogens, it has been believed that Cryptosporidium has multiple multiple species, but the so the same route of transmission, that is by the ingestion of contaminated water or the commodities in which the same water has been used. It is also what
Dr. Weiss, we can't hear you clearly, sir. Can you please check your internet?
Dr. Muhammad Weiss.
Muhammad Weiss, please restart your maybe laptop, some problem in the internet. We can't hear you clearly, sir.
Okay. Meanwhile, we can share the code QR. Okay, until Dr. Muhammad always get ready and solve the problem of the sound from his part. We will share now the code QR for our attendee to get their participation certificate.
Doctor?
So in the chat box, we shared the link.
Okay. So that's it for Dr. Owais. And we have now the link below for anyone that wants us to send them a certificate of attendance for the first session. Just please click on the link below and type your name, and we will send you the certificate in your email. And we conclude that this is the end of the first session and the first day. Thank you very much all for coming and for listening. And we will come back again at 1 p.m. for the second session of the first day of the webinar. Thank you very much.
stop the video. Yes. Dr. Muhammad Awais, please share your content.
Okay,
Yes, please
can
go
you
ahead,
listen
Doctor.
to me now?
Yes, please go
We can
ahead.
hear you clearly,
Okay.
Dr. Muhammad Awais. Thank you.
Yes.
Okay, thank you very much. So, waterborne parasitic diseases. So, the next category is for the entamoeba histolytica infection. That is one of the leading causes of amoebiasis in the world, in the country and worldwide as well. So, the disease is highly prevalent around the globe. It is also known as a disease of poverty because it is mainly associated with the poor hygiene, poor personal hygiene, and the poor socioeconomic status of the individuals living in a society. So it is usually transmitted by ingestion of contaminated water and food commodities. It has worldwide distribution. It might be in two forms, that is in intestinal amoebiasis or extraintestinal amoebiasis. So in intestinal amoebiasis, it may lead to abdominal cramps, nausea, fatigue, anorexia, and other intestinal problems. So in severe case, there may be dysentery, that is the bloody diarrhea, then there may be perfused vomiting and irritable bowel disease. But whenever such patients are not cured, it may lead to extraintestinal amoebiasis in which it penetrates through the intestinal lumen and it go to the lungs, leading to pulmonary amoebiasis. It may go to the liver, leading to liver amoebic abscess, and may go to the brain, leading to abscess development in the brain. So the next one is the Blantidium coli, that is the largest pathogenic protozoa which infects the human beings. It is transmitted by contaminated water and food, and the pigs acts as a main reservoir host. So it is more prevalent in industrialized regions of the world where pig is being raised for food purpose. It leads to GIT disturbances like diarrhea, dysentery, nausea, then tenesmus, vomiting, and anorexia, whereas other signs might include the headache, insomnia, pain in the muscles, weight loss, and colitis. So this is the pictorial presentation for the possible sources of transmission of Cryptosporidium, Giardia, Cyclospora, and Entamoeba species from water bodies to human population. So the contaminated and by the consumption of contaminated water that might be untreated or unboiled, it may lead to the transmission of cryptosporidiosis, giardiasis, cyclosporiasis, or amoebiasis. So in most of the developing countries, the night soil or the sewage water is used for irrigation of different vegetables. So when these vegetables are being used as such, these may contain the cysts or eggs of these parasites, and these may contaminate these vegetables and get and maybe transferred to the consumers when these vegetables are taken as such without proper washing or without proper treatment by the vinegar. So it is strongly recommended that whenever you consume such commodities, so treat it with the vinegar, which is which has strong cysticidal activity. It may kill the cyst and it may be safe to consume. So the parasites which can be transmitted through meat, with these includes the Toxoplasma gondii, Taenia saginata, and Taenia solium. These are transmitted by the ingestion of the undercooked or raw meat of the birds, sheep, goat, chicken, pigs, cattle, and pigs. Then they have variable distribution throughout the world, and then they have the clinical manifestation in the form of GIT disturbances. All this. Then the next one is the Trichinella spiralis and Sarcocystis. These are transmitted by ingesting the meat of the pigs, boar, horse, and cattle, whereas Sarcocystis is transmitted by ingesting the meat of cattle and wild captive animals. So this Trichinella spiralis is worldwide in distribution, especially in Europe and African countries, whereas Sarcocystis is more prevalent in the Thailand, Malaysia, Mexico, African, and African countries. Though clinical manifestations are same like GI disturbances, maybe abdominal pain, diarrhea, constipation, fever, and other similar signs. So these are some other meat-borne or seafood-borne diseases. So will lead to the factors which affect the global incidence of foodborne parasitic zoonosis. These might include the poor sanitary conditions, malnutrition, which made the consumers immunosuppressed, or their immunocompetency is being lost, and ultimately the pathogen upon entering the host, they get develop infection. Then the changing demographics means the increasing population of infants and elderly in different localities. Then inadequate public health infrastructure in most part of the world, especially in the developing and underdeveloped countries of the world. Then inadequate hygienic and technological conditions of food production and processing. Then inadequate cooking, reheating, and storage conditions also cause the transmission of these diseases. Then increasing tourism and international trade without any appropriate SOPs. Then increasing animal movement and insufficient control of borders, increasing international trade of animal and food, inadequate legislation and official control system like HACCP, then emerging and re-emerging foodborne pathogens, adaptation and enhanced survival of pathogens in food, and the most important is the inadequate consumer education about the safe food. So what are the zoonosis associated challenges? World has badly struck with the zoonotic infection, and these are a big challenge for medical and veterinary sectors as follows, like zoonosis versus socioeconomic instability. So these zoonotic pathogens, they impose a ban on international trade, which badly affect the trade of animals and their products from one country to the other. And it is a big problem in the developing and underdeveloped countries, thus causing a great loss to the in terms of the poor revenue generation. Then economy of the country, for having the negative impact on industries. Then social isolation, countries which are being reported for the presence for being endemic for certain zoonotic pathogens, they are being isolated from rest of the world, and their animals or their animal products are being not exported or imported. Then zoonosis versus public health concerns. The zoonotic pathogens, they pose serious threats to veterinary and medical sectors. So they cause high rates of deaths and disease, means morbidity and mortality among the human population. Then domestic or wild animals, they are responsible for 75% of the emerging diseases which are being originated from these domestic or wild animals. So what are the critical gaps in opting the One Health approach? So what is the One Health approach? That is to bring all the health professionals like the physicians, like the veterinarians, like the food microbiologists, like the epidemiologists, like the disease diagnostic personnel, like the agriculturists under one forum to find out the solutions to control these diseases at their own parts, because a veterinarian alone cannot rectify the condition, or a physician alone cannot rectify the problem. So this is an intercoordinated approach which can help us to put a full stop in the spread of these zoonotic diseases. So the limited ownership for zoonosis prevention and control by any one of these sectors is the major problem. So there is no specific contingency plan for the control of these zoonotic diseases. Inadequate surveillance system in most part of the world is other reason. Then limited strategic stockpiles of life-saving drugs in case of the outbreaks of such zoonotic pathogens. Then infrequent coordination among human and animal health sectors is the most important factor here. Then insufficient resources for surveillance and response activities against zoonosis. Then non-involvement of private sector veterinary health facilities in the absence of legal framework for national and international disease surveillance programs means we don't have any coordinated surveillance system at national and international platforms. Then there is a lack of any dedicated body organization at local level to coordinate zoonotic disease surveillance and response activities focusing One Health. But the encouraging thing is now people have started thinking on this particular approach, and most of the funding agencies and most of the organizations on working on to control such diseases in a One Health perspective. So how we can prevent and control? So as I mentioned that there is a great concern about the zoonotic diseases at 3 different levels, including the consumers, the manufacturers, and the policymakers. So at consumers levels, they should adopt appropriate hygienic measures, like they should rectify their feeding practices, cooking practices, and handling the potential food reservoirs to control the transmission of zoonotic pathogens. Then the manufacturers, they should ensure the hygiene of food handlers like hostels and restaurants. Then proper screening tests of potential food commodities for parasites before marketing to avoid the spread of these pathogens in the human population. Then the policy making bodies, these includes the official and administrative authorities. They should develop a stringent public health system. Then they should develop the surveillance and epidemiological studies for early diagnosis and early response to the outbreaks of these diseases. And then the most important is to develop the collaboration and coordination with the academia and other research organizations for understanding the disease patterns. So what are the way forwards towards strategic control of zoonotic diseases? So understanding our expectations of emerging pathogens will help us design the active engagement and collaboration between zoonotic disease experts in public health and animal health sectors, that is IHR mechanism. Then the effective surveillance programs should be run. That is the first line of defense, making rapid detection and identification of key priorities. Then effective international cooperation, which includes the timely sharing of data and biological specimens, effective communication and cooperation between those working in human and animal health sectors. Then we should go for strategic planning for vaccine production through local isolates and development of isolate reserve bank at country or regional level, because most of the time when we use the vaccines, the serotypes are the or you can say the antigenic determinants of the pathogens used on these vaccines do not match with the isolates of the pathogens which are prevalent in our countries. That is a big reason of vaccine failure. Then we should use multidisciplinary approaches in which clinicians, veterinarians, researchers, public, environment scientists, and global concerns should be considered. Then separate risk analysis units should be established, which may be helpful in estimating the disease burden. Then health institutes, universities, and laboratories should be at one line to take any task for the exploration of a disease incidence, then efforts should be made to develop primary health care across the country. A lot is yet to be done, so we should seriously think on it. Then listing of national notifiable diseases and zoonoses, and there should be more funding for these diseases to tackle. Then we should pursue the regional and international organization for participating in their One Health related activities. So in short, or in nutshell, the key to prevention is strong intersectoral coordination at all levels to control disease, then first-hand response planning at national level, then sharing of information between relevant surveillance systems for management and knowledge of zoonotic diseases, and to improve the global health and decrease burden of communicable diseases. So what are the conclusions? The foodborne parasitic infections are a major threat to human health. They cause huge economic losses throughout the world. Then the developing and underdeveloped countries are more prone to foodborne parasitic zoonosis, predisposing immunosuppression and secondary diseases. They predispose the immunosuppression, which leads to the development of secondary disease conditions. The key level of control are at consumer, manufacturer, and administrative levels, which should be taken into account while devising the prevention and control strategies. So what are the recommendations? So there is a need for the adoption of hygienic measures, monitoring of food handlers, adequate cooking should be ensured, water decontamination, municipal authorities before consumption, improved coordination among public health and veterinary health, environmental health, and food safety services. Then initiatives should be taken for biological control programs. So now a little brief introduction about my university, that is Bahauddin Zakariya University, located at Bosan Road, Multan, Pakistan, which is a progressive university of the country, which was established in 1975. So its main campus is spread over 960 acres of land, which has 2 sub-campuses, 8 faculties, including 4 imparting professional education, including veterinary sciences, engineering sciences, pharmacy, and agriculture. It has 51 departments, institutes, and colleges. Faculty strength is around 700, and more than 40,000 students have been enrolled, out of which more than 50% are female by gender. More than 120 undergraduate and postgraduate programs are being offered by the university. So this is my faculty, that is Faculty of Veterinary Sciences. So these are some of the previous events on zoonosis which we have earlier conducted. The first international conference on zoonosis that was conducted in 2014. Then the second one in 2016. So these are the research activities underway at One Health Research Lab, that is my lab at Department of Pathology. We are working on molecular profiling of zoonotic enteropathogens at animal-human environment interface in Cholistan Desert, a way to formulate veterinary and public health policies through One Health approach that has been funded by Higher Education Commission, Government of Pakistan. And the second one is the sero and molecular epidemiological studies on Brucella species circulating in ruminant and human populations of southern Punjab, Pakistan, a One Health perspective. This project is funded by Agricultural Linkages Program, Pakistan Agricultural Research Council of Pakistan. So this is my research team. So these are few glimpses of preliminary survey to the Cholistan Desert. You can see the water which they are using for drinking purpose. These are the water reservoirs from where they are drinking the water. These are the same source from where animals and human beings are intaking the water. So this is their lifestyle, and the animals are drinking water from these water bodies. So thank you very much. If you have any questions, you are more than welcome.
Thank you very much, Dr. Muhammad Awais, for talking to us about the significance of foodborne parasitic zoonosis. And you have also shared with us that strong intersectorial coordination is required to minimize the zoonotic infection. I have just one question. Yes. Can we say that COVID-19 is also a zoonotic infection?
Actually, there are no systematic studies which have been conducted to find out this COVID-19 as a zoonotic problem. Although some of the organizations have made some serological studies on the detection of this COVID-19. Basically, this COVID-19 virus is not a new virus. In poultry, we already have a disease that is infectious bursal disease that is caused by coronavirus. So this is basically a variant of the coronavirus. Basically, it has been originated from the animals, as you might have listened about the history of this COVID-19 virus that has been emerged from the snake and the bats by the development of mutation. So as right now we are fighting hard to control this disease in human population, but at the same time people are also working on the exploration of the possibility to find out either there is a zoonotic pathway is there or not. So at right now we cannot say anything in a very specific manner, but yes, coronaviruses do exist in animals. They do cause different diseases in animals. So the zoonotic link is being established right now, but there is no authentic report yet.
Okay, thank you very much, Dr. Muhammad Awais. Yeah, any question from the audience, please? Okay, thank you very much, Dr. Muhammad Awais, for talking.
Thank you very much, Dr. Muhammad Awais.
Thank you very much to the organizers for providing me the opportunity to talk at this prestigious event. So thank you very much to all of you. Thank you.
Thank you very much. So we take a break and we return at 1 p.m. KSA time. Okay. Thank you very much.
Thank you.
Assalamu alaikum wa rahmatullah. Welcome back to the second session of the first day, the second international webinar on science, sustainable development and ecosystem in Saudi Arabia. We will have the first speaker, Dr. Isam Qutb. He is an associate professor of microbiology at the College of Science, Imam Abdurrahman bin Faisal University. He is especially concerned with the potential of microbe enzyme in the distillation of different environment and medical polymers, such as fibrin. He wrote a book and he wrote many papers in high-impacted factor journals. He is also the head of biotechnology unit and in the Basic and Applied Scientific Research Center in Imam Abdurrahman bin Faisal University. I welcome my colleague at the department, Dr. Isam.
Thank you, Dr. Al-Mashir. Thank you. You hear me?
Can you hear me, Dr. Isam?
Yes, doctor. You hear Dr. Al-Mashir? Doctor? Assalamu alaikum.
Assalamu alaikum. Yes, we are hearing you, Dr. Isam.
Yes, Dr. Al-Mashir not hearing me?
No, she's hearing you.
Okay, doctor. Start?
Yes, please.
Yes, okay. Good afternoon. First of all, I want to thank the organizers for giving me this chance to share this research with you. I want to thank Dr. Iman, Dr. Rida Hamdi, and Dr. Al-Mahshir, the chairperson of this session. I am especially interested in the field of bacterial enzymes and their potential in the degradation of some compounds like fibrin, chitin, keratin, phosphate, and oxalate. My talk will be about the integration of contaminating, large screen, integration of the contaminating oxalate from the environment into the food chain and the role of oxalate decarboxylases from biotic bacteria. We know that a variety of salts can be crystallized from the environment inside the human body. When your urinary system tries to excrete them out, under low pH of the urine, oxalate may be precipitated anywhere in urinary system as the kidneys, ureters, or bladder, forming what's called urinary tract system stones. In fact, these oxalates are naturally found in a variety of foods, including certain fruits, vegetables, especially the dark green vegetables like spinach, beans, nuts, grains, many, chocolate, beer, berries, many foods containing these oxalates. Till now, oxalate don't have any problem because it's present in the form of monovalent metal. It's found in the form of potassium oxalate, sodium oxalate. The problem comes when the potassium oxalate or sodium oxalate from your diet coupled with the free calcium in your body fluids, in the urine or blood. Calcium oxalate will be formed by simple replacement reaction between calcium and potassium or sodium oxalate. In fact, the kidney stone disease affecting is or life risk for 13% of population, especially the adults, because the adults have free calcium in their body fluids where the reaction can occur. While children or infants, all the calcium go to build their skeletal system, so they don't have this disease. All people tend to develop these crystals in the form of sand-like material, gravel, stones embedded in protein complexes. The most abundant are stones made up of calcium oxalate, which means that most of stones in our bodies are calcium oxalate. More than 87% of stones are calcium oxalate, while the rest of stones are made up of calcium phosphate or urate. In parallel, calcium oxalate is also formed in the environment by some fungi, especially the litter fungi, or what we called it as litter fungi. Some litter fungi, in response to environmental pHs, they produce calcium oxalate. For example, we know that fungi prefer the acidic pHs for living, for growth and metabolism. If the environment rises from the acidic pHs, special types of fungi produce calcium oxalate to minimize the pH again. If neighboring bacteria is present in the microenvironment of these fungi, we know that bacteria prefer the neutral condition. Some of these bacteria are able to degrade the formed oxalate produced by fungi. Bacteria can degrade oxalate by two types of enzymes. The first one called oxalate decarboxylase, and second one is called oxalate oxidase. The difference among them is that oxalate decarboxylase degrade calcium oxalate to formate and carbon dioxide, while oxidase is much more potent. It degrades oxalate to carbon dioxide and the hydrogen peroxide. In our lab, or any lab, you can determine the type of enzyme produced by measuring the end product from these reactions. For this, oxalate decarboxylase and oxidase are very important for improving our health. and removing such type of crystals. Referring to literature, oxalate decarboxylases are discovered or were discovered in many fungi, especially genus Cordyceps, genus Aspergillus, genus Trichoderma. Many Aspergilli, Microspora, Cordyceps can produce this enzyme. Also, some bacteria can produce oxalate decarboxylase, like Bacillus subtilis, Oxalobacter formigenes, Streptococcus faecalis, Proteus rudgeri, and Elantum, in addition to Escherichia coli. If you read in literature, you will find most of paper focused on Oxalobacter formigenes and Oxalobacter oxalaticus. These are the most important bacteria in literatures. For us, we make a screening program for lactobacillus bacteria or lactic acid bacteria, and detecting the ability of these bacteria to produce oxalate decarboxylases. Lactic acid bacteria is known as probiotic. They are very safe for human. You can consume the bacterial cell with no problem. We isolated several lactobacilli on demand rugosa and SARS agar, which is called MRS agar. This is a specific medium for lactic acid bacteria. The isolated bacteria on MRS agar were checked for enzymatic productivity on a specific medium for oxalate degradation called oxalate clearing medium or OCM medium. This medium is formulated in a special manner. We formulate agar containing potassium oxalate and several minerals as precursors for bacteria to produce the enzyme. After sterilization and solidification, you should rinse your agar with calcium chloride to make the replacement reaction and forming artificial calcium oxalate in your agar. Now, you can test the bacteria against the calcium oxalate in your Petri dishes. Depending on this, we isolated many bacteria degrading oxalate. The most potent or the most powerful strain was strain KSK2, isolated from an Egyptian fermented food, which is a traditional food in Egypt called kishk. This bacterium was the most potent isolate belonging to my isolate. Identification or species characterization was done according to the main key for bacteriological identification, which is Berg's Manual of Systematic Bacteriology. Then species characterization and confirmation was done using the API kits specific for lactobacillus, which is API 50CH. Go to the next experiment. When incubating our strain in the production medium, we initially found that bacteria produce the enzyme at 6 pH, while any change in the pH, even neutral pH, declines the production. It was very sensitive for any change in the pH. While tracking the decarboxylation or the enzyme production by time during fermentation, we found that bacteria is lagging during the 24 hours. You see my mouse? See my mouse, doctor? I am pointing.
Yes, yes, we can see you.
Yes, yes, doctor.
Go ahead, please.
Okay.
Yes, Dr. Hassan.
Okay, we see lagging in the decarboxylation of oxalate during the 24 hours. So bacteria need more time to decarboxylate or degrade the oxalate in the medium. It degrades the oxalate very well after the third day, after 72 hours, with percent reaching 80% of oxalate was degraded. We tracked the oxalate degradation by two methods. The outcomes for mate assay kit purchased from Sigma and measuring the residual oxalate by titration against potassium permanganate. While the bacterial growth was optimal at the second day, in the field of enzymology, we call this a secondary metabolism. The bacteria is building a secondary metabolite, not a primary metabolite. So this enzyme could be concluded that it produced during the stationary phase of bacterial growth after the third day. During my experiments, I noticed that no enzyme production in the complete absence of oxalate. In the field of enzymology, this is a rare phenomenon. In most enzymes I have worked, enzymes are constitutive. They can be produced even the substrate is not present. This enzyme must be present in the medium for induction. We call this is inducible enzyme, not constitutive enzyme. Regarding the effect of carbon and nitrogen sources on the oxalic carboxylase production from the tested bacterium, we found conflicting results. The simple carbon sources or the simple sugars like glucose, fructose were best for enzyme production in comparison with complex polysaccharide and disaccharide. When you see the number of units with glucose and fructose, you see it's more higher than the complex carbon sources. In contrary, nitrogen sources, the simplest nitrogen sources, which are the inorganic nitrogen sources, like potassium nitrate, ammonium nitrate, were inferior in enzyme production when compared with organic nitrogen sources like soybean yeast, casein, and the amino acid alanine and glutamic acid. Here we can conclude that the complex nitrogen were better for enzyme production, while the simple sugars were better for enzyme production. We fixed this in our medium to optimize the final medium for enzyme production. As we study a probiotic, you should put in your mind to study the effect of prebiotics. Prebiotics are non-digestible fibers or non-digestible polysaccharides that your body not digest. These fibers are very important for our health and also for bacteria. These bacteria prefer prebiotics or these fibers. We tested the effect of them at 0.1% concentration, and we found that arabinogalactan and fructooligosacchariide, FOS, you can find this in supermarkets now. It's become commercial. Fructooligosacchariide also found also was good for enzyme production from the bacterium, while inferior production was obtained with Arabic gum. But in all cases, all the tested prebiotics were better than their absence as compared with the blank treatment or the control treatment. Now we finalize the production medium after adjusting the physical, nutritional factors affecting the bacterium. Now we cultivate about 3 liters of the culture medium for enzyme separation and purification. Enzyme separation followed this scheme, which starts from ammonium sulfate precipitation at fractions 30 to 60% ammonium sulfate fractions. Precipitated proteins with ammonium sulfate were dialyzed against buffer, then eluted through diethyl aminoacyl separase, which is the ion exchanger. Active fractions from the first column were pooled, lyophilized with the lyophilizer, then eluted through the second column, which is a gel permeation column or the Cefadex G100 fast flow column. We obtained a recovery of 14.8, and the purification folds were increased to 19.6 with this scheme. The final eluted proteins from the last chromatogram through Cefadex G100, the active fractions formed a major peak between fraction 27 to fraction 35. These fractions were collected or pooled and lyophilized for SSDE page analysis or the vertical electrophoresis study. We obtained a major band on this land, which is indicative of the molecular weight at 63 kilodalton. The presence of one band in this land is indicative of pure enzyme. When we make agar supplemented with potassium and rinsed with calcium, with micro wells bunched off with the capillary glass tubes, and filling these wells with only just 6 microliters of purified enzyme, we obtained good results. This is the replicate within 12 hours, only 12 hours, which means that your enzyme, when become more purified, takes less time. Concerning the biochemical properties of my enzyme, we studied both pH on activity and stability and temperature in both activity and stability. We found that the enzyme was stable between pH 4 to pH 7, and best pH for enzyme activity was at pH 5 and pH 4.5. pH stability measured by incubation of enzyme alone for 1 hour, then add substrate. While for testing the pH, effect of pH on enzyme activity, you incubate both enzyme and substrate at the beginning. This is the difference between studying the stability and the activity. The same for temperature. We found that the enzyme was thermally stable below 70, and it was most active at 35 degrees centigrade. This is normal pattern for Cuban enzyme superfamily, from which this enzyme belongs because of the large number of subunits, short lobes, efficient packing, hydrophobic interactions, hydrogen bonding, and the fewer cavities of this enzyme. Now we move to the effect of some reagents or some chemicals on the enzymatic activity. We choose these chemicals or reagents based on literature. These chemicals, some of them are found in our body fluids, some are found in the clinical laboratory during the measurement of oxalate from your blood. So we want to test the compatibility of enzyme with these chemicals to prepare ourselves for application. So the compatibility of enzyme with these chemicals is very important for us. We found that sodium ascorbate, potassium ferrocyanide, and ODA, which is orthophenylene diamine, these compounds increase the enzyme activity. While the stronger reducing agents like orthohydroxylamine, sodium diocyanate, sodium sulfite decrease the enzymatic activity. Using the APCAM format assay kit, reagents like Triton X100 increase the enzymatic activity. Sodium halides in the form of bromide, fluoride, iodide, chloride inhibited the enzyme, only chloride not inhibited the enzyme. The effect of carboxylic acids like this group, malic, oxaloacetic, ketoglutaric, succinic, fumaric, malonic, citric, salic, this carboxylic acid from which oxalate belongs, were tested as competitive inhibitors for this enzyme. We found low competitive inhibition of these organic acids on the enzyme activity. This is measured by the assay kits and by this agar plate. Here you see the controlled treatment without the effect of organic acids. Here we find the inhibition or the competitive inhibition of some organic acids like malonic, malonic acid, fumaric acid. This is the highest inhibition are here, while other acids like phthalic, ketoglutaric exerts less inhibition effect upon the enzyme or are weak competitive inhibitors. It is very important for enzyme to measure the enzyme kinetic parameters. We call them Vmax, Km, Kcat, Kcat over Km. The Vmax is the maximum rate of reaction. You want to study where the enzyme goes. Where is the maximum rate? Above which no exceeding in the reaction due to the saturation of the enzyme with substrate. Here from the hyperbolic curve, you will see here the lagging or the stationary phase of reaction at this level, which corresponds to 25 millimolar per minute. This is the maximum rate of reactions. Kinetics is very important to know the dynamics of your reaction. If your enzyme is slow or it is go in reaction with high speed. This is what I mean. Halfway of Vmax called Michaelis constant. This is according to Michaelis-Menten equation. Half Vmax is called Michaelis constant. We obtained the Michaelis constant from the hyperbolic and the Lineweaver bulk plot. Lineweaver bulk plot is the reciprocal of hyperbolic curve. From this equation, you can easily know all these parameters: Vmax, Km, Kcat. Kcat is the turnover number, which is the number of molecules of substrate degraded per minute from the substrate. Kcat, when you divide Kcat versus Km, this is the catalytic efficiency of your enzyme. This is like a fingerprint. It's very important to compare your kinetic parameters with others to know is your enzyme is efficient or less efficient. So we calculate the kinetic parameters using potassium oxalate as a substrate from the hyperbolic and the Lineweaver bulk plot. Finally, we formulate this in vivo experiment with my colleagues in the zoology department. We divided rats into 5 groups. Rats weighing 150 gram body weight. The first group served as a universal control group fed with a standard diet with no treatment, no stress at all. Group A fed with the sodium glycolate. Sodium glycolate is a precursor for stones, plus diet. Group B fed with the glycolate diet plus the tested bacterial cell is a probiotic. Group C, same as B, in addition to a prebiotic mixture, الفركتooligosaccharides and arabinogalactan. Last group is same as the group C, but without the bacterial cell. Just you test the effect of a prebiotic only on the alimentary canal bacteria. Rats were fed in this manner for 10 days. After passing 10 days, urine samples were collected for analysis of oxalate, phosphate, and oxalate in urine samples. Group C, which received both probiotic and prebiotic, we found that oxalate reaches 0.7 when compared with Group B, Group A, control, Group D. Here is a prebiotic only, here prebiotic and probiotic, and so on. From this, we can conclude that the presence of a prebiotic and probiotic mixture, which call it symbiotic. Symbiotic means that both probiotic and prebiotic, both of them, are better for minimizing oxalate level in these rats. Conclusion: human body lacks the enzyme system required to solubilize oxalate brought inside as a constituent of many foods and environmental sample. The gut microbiota in your body have shown to play this role. But under unbalanced conditions, such as oral intake of antibiotics, you kill these gut microorganisms by the intake of antibiotics and other drugs. So losing of this gut microbiota make you at high risk of calcium oxalate formation in your body. In this study, treatment of oxaliuric rats with L. plantarum and the prebiotic mixture, fructooligosaccharides and arabinogalactan, significantly decrease the oxalate levels inside the body. Best results in group C were obtained in the presence of both of them, suggesting a successful symbiotic system for prevention of oxalate stone formation. This enzyme may be also useful in clinical laboratories for measuring oxalate levels in your body because it's compatible with some chemicals as we saw in the table. It's a thermotolerance, activation by triton, less inhibition with the reducing agents and the chloride. So it is a promising enzyme in measuring oxalate in your body fluids. Thank you.
Thank you very much, Dr. Issam, for this interesting presentation. Can you tell us about your future studies?
Doctor, I am now working on chitinase and keratinase. In our unit, in biotechnology unit, we are working on chitinase, keratinase, and phosphatase. For keratinase, we don't obtain good results with this enzyme. But chitinase, we obtained more than 40 isolates degrading crustacean wastes in an excellent manner. So inshallah, I will focus in this year, inshallah, on chitinase and phosphatase.
Inshallah, that's excellent, Dr. Issam. If anyone of the audience...
Yeah, I have a question, Dr.
Yes, please go ahead.
First of all, I want to thank Dr. Issam for this nice presentation.
Thank you.
But since I am from physics department, I don't know so much about this, let's say, biology field. But I have few questions. Maybe they are not so good. But anyway, first question is, how you can measure the enzyme production from the bacteria?
Okay, excellent, doctor. Doctor, now you have substrate, which here is oxalate, okay? Enzyme react with the substrate, giving you a product, right?
Yeah.
So you can measure the activity by two ways: to measure the amount of product released, or measure the remaining oxalate. In this enzyme, you have the two options. You can first you know what you added, the amount of initial oxalate, okay, right? So you can measure the remaining oxalate by titration with potassium permanganate. If we have any chemists here, you know that permanganate, which is potassium KMnO4, react with oxalate by titration. So we can determine the remaining oxalate by titration against potassium permanganate, or measure the formate, which is the end product, by the outcomes formate acetate. Understand me, doctor? So enzyme production or enzyme activity in all cases, in all enzymes, determined from the end product.
Okay.
If you have a product, you have enzyme. No product, no enzyme.
Okay, I got it. Yeah, thank you, doctor. Okay. The second question is, wow, this is really a stupid question, but my question is, if you increase the number of bacteria, you will increase the production of the enzyme? Am I right?
Till certain limit, Dr. Rida. Till certain limit.
So there is certain threshold, yeah?
It's not absolute thing. It's till certain limit, like enzyme. Enzyme at certain limit, if you add substrate, it will not speed the reaction. It will be fixed due to that saturation of enzyme. You know that enzyme have active sites, like key and lock. Enzyme have active sites, so it reacts with substrate according to the number of active moieties or active sites. If the active sites are fully occupied with substrate, after that, any increase in substrate will not increase your reaction because of full saturation of enzyme.
Okay, got it. Thank you. Okay, the last one, but okay. At the end of your presentation, you show us this curve of the kinematic of the enzyme. Is there any model for that?
Yes, we have many models. We have Lineweaver bulk plot. This is the names of scientists, Lineweaver and Bulk. This is a plot of two scientists, Lineweaver and Bulk. Okay. We have many plots. We have Dr. Noureddin, you know Dr. Noureddin from Tunisia.
Yeah, I know him, yeah.
Yes, he did a plot, a special plot for this.
Because it seems like, let's say, a charge of the capacitor in physics.
Yes, doctor, it's absolutely modeling.
Okay, thank you again for your nice presentation. Thank you again.
Thank you, doctor.
Thank you so much again, Dr. Isam, for this interesting talk.
Thank you, doctor.
And we will move. Dr. Rida already asked the question. And we will move to the next speaker. But before we move, I just want to remind everybody that if you want to have a certificate of attendance, at the end of the second session, we will have a link. And just please click on the link at the end of the session, so we can send you an email with the certificate. Now, our next speaker, Dr. Zafir Hashim. He is an assistant professor at Department of Chemistry, Cosmostas, University, Islamabad, Pakistan. He received his PhD in environmental engineering from Xi'an University in China. He has research interests to develop treatment technologies to control environmental and industrial pollution, to understand toxicology mechanism of PCBs, and to develop novel method for monitoring of environmental pollution. He will speak to us today. Dr. Zafir, are you with us?
Thank you. Thank you very much, Dr. Hanan, for your kind introduction. Can you listen me?
Yes, yes. If you can speak just a little bit louder.
Okay. Thank you. Thank you very much, Doctor, for your kind introduction. First of all, I want to congratulate organizing committee for organizing the second international webinar on science and development ecosystem in Saudi Arabia. You are doing very excellent job, and I want to congratulate all of you. Further, I honor and gratitude that I will deliver my talk in your webinar. Is it fine?
Yes, doctor.
The title of my talk is Polychlorinated Biphenyls Congener-Induced Toxicity in Human Lung Fibroblast Cell Lines. These are the contents of my talk, the structure-dependent 4-chlorinated cytotoxic effects to HELF cells and potential molecular mechanisms. Biphasic dose response of PCBs with 5-chlorination exposed to HELF cells and potential molecular mechanisms. As you know that in toxicology and in pharmacology, dose response studies are very important because from these dose response studies, one can easily figure out standards and threshold levels for chemicals if we want to make some drugs commercialized. First of all, the scientists check the dose response studies of that drug. In toxicology, two models are generally accepted. These models are old models, and the first one is the threshold model. And according to this model, when a dose is applied to a certain endpoint, then at some level, the dose and the response remains the same. However, when the dose becomes very high, then the response of an endpoint also becomes very high. This model is known as the threshold model in toxicology. Another model is linear non-threshold model, and according to this model, when a dose of some chemical is applied to some endpoints, then as the dose increases, then the response of that endpoint also increases. These two models are old models, and the scientific communities consider these two models false, blunder, and these models are not reliable. However, in recent years, a new model has emerged, that model is known as the not working. There's some problem. Just wait, actually the slide is not moving.
Take your time, please, Doctor.
Just wait a minute. Yes, another model is a biphasic model, and according to this model, that when a dose of some chemical is applied, then at low level of that doses, the response of the endpoint become maximum. While when the doses become higher, then the response also decreases. So this model is considered more accurate. It is considered as robust and highly generalized model. So the question is arising that whenever we have to do toxicity studies for some chemicals, then what are the characteristics of different endpoints which we choose to determine the response of a chemical? There are some characteristics of the endpoint, and there are some characteristics of these models. These are highly generalizable, independent of biological model, independent of endpoint, independent of inducing agent, and gave some accurate prediction. There are some biological endpoint has been studied to figure out the toxicity mechanism, and these have been already reported in different literature, such as some scientists study the behavioral responses with respect to some chemicals. They also studied the metabolism, the chemical effects on the metabolism endpoint. They use some bacteria and check the luminescence in the bacteria, and also they check the chemotaxis for some chemicals. Some studies have been also done for lifespan and longevity. However, there is another model which is known as growth, and there is a lack of research about this endpoint for some chemicals to figure out the toxicity mechanism. This model is studied. It is robust and highly reproducible. So it is very important to understand the toxicological mechanism using biphasic dose response model for the growth of any cell or any animal. As you know that electronic waste has become a significant environmental issue. Upon incineration and recycling of the electronic products, the electronic waste produce some chemicals. These may be polybrominated diphenyl ethers, some heavy metal, and some polychlorinated biphenyls. As you can see that the structure of PCBs, this is the structure of PCBs, they are highly used in transformers, some electronic products, and these PCBs have 209 different types, and they have some characteristics such as they are hydrophobic, bioaccumulative, recalcitrant, and toxic. They have been produced in USA 6 multiplied 10 raised to power 8 kg. In China, they have been reported PCBs has been reported 1 multiplied 10 raised to power 7 kg. They sometimes the human beings sometimes they get expose these chemicals through inhalation. Sometimes they also get exposure through the dietary, through food chain. Once they enter into the human bodies, they pose some effects to the humans, such as tumor production, dermal toxicity, hepatotoxicity, immunotoxicity, carcinogen, reproductive effects, and neurotoxicity. PCBs, they have some generalized pathway. They are persistent organic proteins. They migrate and persist in the environment. They have some effects to the grassroots level. Sometimes they are volatile, and sometimes they also transfer in the food chain, and ultimately they pose some effects to the human being as the top in the traffic level in the food chain levels. The studies regarding the PCB's toxicity mechanisms are lacking. There's no research and no one knows that what are the exact mechanisms of PCB's toxicity. It has been reported that there are some cell signaling mechanisms, and these mechanisms are reported for some PCBs, such as for wheel cell. The scientists have just developed some potential molecular mechanisms to understand the toxicity, and these mechanisms are considered as highly authentic mechanism for cell proliferation, for apoptosis. And there is another mechanism. These are known as receptor and overcompensation mechanisms. They are less considered, and they are considered as unreliable, and we cannot figure out the exact toxicity mechanism through these mechanisms. So when the chemicals enter into the human bodies, the question arises that what are the exact mechanisms of these chemicals? Basically, when the chemicals are exposed to the cell, first of all, the reactive oxygen CPC is activated, and there are different mechanisms. There are different oxidant and antioxidant enzymes. They are activated, and they can trigger some proteins. These proteins also activate the cell signaling pathways. So there is a lack of research regarding the PCBs trigger reactive oxygen species, oxidant and antioxidant enzyme response. And after that, the responsible mitogen activated protein kinase the pathway for after exposure to PCBs. Human lungs receive some chemicals to inhalation and to food chain. When we categorize the PCBs have 209 different types. Some are low chlorinated. These low chlorinated PCBs are volatile. Sometimes the PCBs are high chlorinated. They are not volatile, but they move in the atmosphere, in the environment through food chains. So there is a lack of research regarding the PCB's toxicity in the human lung cell. So I generated idea that if we choose human lung cells, then what are the molecular mechanisms and how the polychlorinated biphenyls, this is a E-based pollutant, how this activates cell proliferation and human lung fibroblast cells. Why I choose the human lung fibroblast cell? Because the human lungs or any lungs receive chemicals through inhalation and through blood through food chain. So these lungs are highly vulnerable to the chemicals. So these were the objectives. I choose low chlorinated and high chlorinated PCBs, and then I check the response of these PCBs on cell proliferation. I use some proteins, some oxidative stress, antioxidant response, and potential molecular mechanism. This is the technical roadmap of my study. I choose 4 chlorinated PCBs. You can see here PCBs 40 for this study, then PCBs 77. First of all, I studied the effect of these PCBs on cell proliferation. Then what are the responsible mechanisms behind this cell proliferation? What are the some proteins that express? I studied the apoptosis, membrane integrity, cell cycle. For the cell cycle mechanisms, I studied the cyclin E, CDK1, caspases, and P21 proteins. These are the proteins, and these expressed during the cell cycle and cell proliferation. And these are the responsible proteins after exposure to the chemical PCBs. Then I choose another chemicals, the 5 chlorinated PCBs. And these are PCBs 101 and PCBs 118. I studied the effect of these PCBs on cell proliferation, oxidant and antioxidant response, apoptosis, cell signaling mechanisms, and the cell signaling in the cell signaling mechanism. I studied the ERK1 protein and GNK protein. These are the proteins which are responsible for the cell proliferation and for the cell growth in human lungs fibroblast cells. This is a technical research design. These are the non-coplanar, coplanar, non-coplanar, coplanar PCBs. These are the structure. These have the 4 chlorine atoms, and these PCBs have the 5 chlorine atoms. These are the human lung fibroblast I culture in the lab, and these are the culture conditions which I used during my experiment. So the structure-dependent 4 chlorinated effects on the cells, and these were the technical roadmap for the 4 chlorinated PCBs. These were the different doses which I choose. These doses were considered as triplicate. 3 times the experiment was run to conduct the analysis. This is the experimental methods, HELF cells. These were cultured into CO2 incubator at 35 centigrade and 5% carbon dioxide. These were some empty test, flow cytometry test. Then I did some protein extraction, and these analyses were conducted on microplate reader, flow cytometry, Western blotting, and so on. The results of 4 chlorinated PCBs exhibited that at lower concentration, PCBs 40 induce the cell proliferations, and in HELF cells. These doses were at 10 raised to power minus 5, 10 raised to power minus 3, and 10 raised to power minus 2. These doses significantly enhanced the cell proliferation, stimulated the cell proliferation. The cell proliferation enhancement as compared to control was 104 to 109% as compared to control. So the results suggest that these level could be considered as threshold level or standards to find the risk assessment of PCBs in the environment. PCBs 40. And the response was 108 to 109. The maximum response inhibition for HELF cell proliferation showed that 10 raised to power 2 minus 2 and 10 raised to power minus 5 significantly stimulated the cell proliferation. However, the higher concentrations such as 10 and 20 microgram per ml significantly reduced the cell proliferation. The reduction in the cell proliferation was 4 to 39.97 with the passage of and with respect to higher doses. And these doses, however, after 20 days and 74 hours, maximum reduction was recorded for PCBs 40. Similarly, the results for PCB 77, it indicated that 10 raised to power minus 1 showed maximum stimulation in HELF cell proliferation, and it was 8.5% as compared to control. This is considered as the threshold level for PCB 77 to determine the environmental risk assessment of e-waste pollutant in the environment. However, at a higher concentration, after 72 hours, PCB 77 significantly inhibited the cell proliferation, and it was 45% as compared to control. Then I studied the cell membrane with respect to PCB 40 and PCB 77. 40, these are 4 chlorinated PCBs and non-coplanar. The membrane integrity increases over the time and with the increase. However, PCBs 77 showed that for some doses, the membrane integrity response increased. However, when the doses become higher, then the response of the HELF cell membrane integrity decreases over the time. Then I studied the cell cycle after exposure to PCB40. This is the PCB's concentration, and these are some response after 24 hours, 48 hours, and 74 hours. In the cell cycle, I studied the S phase, G1, G2, and G1 phase, G2 phase, and G1 phase. So the result indicated that the one phase increased after exposure to PCB40. You can see here the maximum expression of the G1 phase over the time was recorded after exposure to PCB40. However, the S phase and the G2 phase decreased, show decreasing trend 2 hours after exposure to PCB40 concentration. So the effects of PCB77 on cell cycle indicated that the G1 phase after 24 hours increased. But after 48 hours and after 74 hours, you can see here the G1 phase decreased, but S1 phase increased. So, and also the apoptosis also increased. So the results suggest that PCB77 are more toxic to cell cycle of HELF cell as compared to PCB40. Then I studied the effects of 4 chlorinated PCBs on the cell cycle protein expressions. Different cell cycle proteins were studied, such as cyclin E, cyclin D1, CDK1, P21, caspases, and this is the beta actin. This was used as a control. The expression of cyclin E, cyclin D1, P21, and caspases 9 decreased after exposure to PCB, while the expression of CDK2 was unchanged as compared to the control. The highest protein contents of cyclin E, you can see here that cyclin E showed highest content were 0.051 for 0.001 microgram per ml. You can see here 0.01 ml, and here the expression of cyclin was maximum. For 0.03 for 0.01 microgram ml, CDK2 were 0.059 for 1 microgram per ml, P21 were 0.033 for 0.01 microgram ml, and caspases were 0.048 for 0.0 microgram per ml of PCBs after 4 hours. The results suggest that cell cycle and apoptosis proteins were less affected by the PCBs. So as we can see here that in the cell cycle flow cytometry results and in the cell proliferation results, the PCBs were less toxic as compared to PCB77. Then the results for cell cycle protein expression for PCB77, and the results indicated that cyclin E, CDK2, and P21 showed increasing trends, while CDK1 and caspases protein showed decreasing trend after 24 hours. PCB77 exposed to HELF cell indicated highest protein content for cyclin E, cyclin D, CDK2, P21, and caspases 9. The results suggest that cell cycle and apoptosis protein were significantly affected by PCB77. If we compare the results of PCB77 and PCB40, so from the results and from the Western blot analysis, we can easily understand that PCBs were more toxic as compared to PCB40. So in conclusion, that Non-coplanar and coplanar PCBs are at non-cytotoxic concentration increase the cell proliferation, while at higher concentrations it decrease. Low concentration of PCBs 40 alter membrane structure, and increase in membrane fluidity suggests that PCBs 40 followed the mitochondrial pathway to kill the HeLF cell. Flow cytometry revealed that PCB 40 increased the percentage of G1 phase, but decreased S phase at concentrations and time-dependent manner. Whereas PCB 77 exposure decreased the percentage of G1 and S phase, but increased G2 phase. Western blot analysis indicated that PCB 77 PCB40 increased the expression of CDK2 and P21. Caspase's 9 activity was found in both PCB congeners, suggest that apoptosis was caspase dependent. Cytotoxic mechanisms of the PCB's congener depend on polarity and chemical structures. So PCB70 was planar. The PCB70 was considered as more toxic as compared to non-coplanar PCBs40. Future perspective for the studies are caspase's protease and P53 should be considered in future cell cycle. The all membranes of cyclin and CDK could be considered in future research. For these chemical oxidative stress effects on cell activities should be investigated. Activation of constitutive androsten receptor CAR after exposure to coplanar and non-coplanar PCBs congeners should be researched further in relation to MAPK pathways. So the result for 5 chlorinated PCBs. This was the experimental design for the 5 chlorinated PCBs. These are the different doses for PCBs 101 and 118. This was cell culturing in the CO2 incubators. MTT, RAS, oxidative stress, anti-oxidative stress, and some proteins were extracted. The MTTS analysis was done in macroplate readers. RAS was also done in macroplate readers. Fluorescent microscopy was used for reactive oxygen species. Some protein analysis was done in Western blotting analysis. So these are the results after exposure to PCBs 101 and 118 on HeLF cell proliferation. The results indicated that PCBs 101 at lower concentrations such as 10 raised to power minus 3 and 10 raised to power minus 1 stimulated the HeLF cell proliferation, and the stimulation was 108.8% to 109.8%. So the results suggest that these could be considered as threshold and standard for to determine the risk assessment of PCBs 101 in the environment. And this was this stimulation was over the time. However, the higher concentration, such as 5 microgram, 10 microgram, and 20 micrograms, the HeLF cells showed inhibitory responses after exposure to PCBs 101. And the inhibition of HeLF cell proliferation was 3.2 to 46.03% over the time. However, the maximum inhibition or reduction in cell proliferation was found after 48 hours as compared to control and as compared to 4 hours, 12 hours, and 24 hours. Similarly, the results indicated for PCBs 118, and you can see in the bar graph that after exposure to PCBs 10 raised to minus 2 and 10 raised to minus 1, the cells showed maximum stimulation as compared to time over the time. And the stimulation of the cell proliferation was 109.8% to 109.9%. So the results suggest that these levels could be considered as threshold level to calculate the environmental risk assessment of PCBs 118 in the e-waste recycling areas. However, the results for the higher doses indicated that 10 microgram per ml and 20 microgram per ml exposure or concentration of PCB 118 decrease this HELF cell proliferation after 48 hours as compared to control and as compared to other times. The reduction in the cell was 1.3% to 71.01%. If we compare the results of both PCBs, you can see here that PCBs 118 showed maximum reduction. The maximum reduction in the HELF cell was 71%. However, the PCBs 101 showed only maximum reduction 46%. So that PCB 118 was more toxic to human lung fibroblast cells as compared to PCBs 101. So I tried to figure out the exact molecular mechanisms. To study this, I studied the activation of oxidative stress exposed to PCB 101 and 118. The reactive oxygen species expression increased for both PCBs over time with respect to higher doses. Then I studied the activation of antioxidative enzymes, and for this I studied the MDA. MDA, this is the antioxidant enzymes to neutralize the effects of reactive oxygen species. So you can see here that antioxidant expression increased for both PCBs at higher doses over the time. So the results suggest that to neutralize the effects of reactive oxygen species, the antioxidant showed maximum expression over the time. Then I studied the oxidative enzymes, and you can see that for this GSHPX enzymes was studied. And you can see here that the expression of this enzymes decreased for both PCBs over the times. Then another enzyme, oxidative enzyme, was also studied. It was SOD enzymes. And you can see here that SOD enzyme showed irregular trends. For lower doses, this enzyme showed maximum expression. However, at higher doses, the expression of SOD enzymes decreased as compared to lower doses. You can see here that at 20 microgram per ml of PCBs, both PCBs, the expression of SOD enzymes is decreasing. Then I studied the genotoxicity, the genotoxic effects of both PCBs. So you can see here that PCBs 101 is less toxic, and tail monument, tail length, DNA tail length, DNA tail contents were less as after exposure to PCB 1. And I took two doses. One is low dose and another the high dose. If we compare the results of PCB 101, then you can see here that PCBs 118 have more genotoxic effects. As you can see here that the tail, long tail of a DNA, and the tail length is also increasing with respect to the higher doses. The results suggest that PCBs 118 was genotoxic as compared to PCB 101. Then to figure out the exact molecular mechanism, that what are the proteins which are responsible for the cell proliferation in HELF cells. Then I studied the mitogen activated protein kinase pathway. So you can see here that after exposure to PCB 101, the expression of ERK protein was prominent after 4 hours and after 48 hours. You can see here the bands that ERK protein, this is the protein which is responsible for cell proliferation. So the results suggest that PCB 101 have less toxic effects on the ERK1 protein. If we compare the results of PCBs 118, so you can see here that with the passage of time and with the passage of doses, with the increase of higher concentration of PCB, so you can see here that after 4 hours, with the increase of concentration of PCBs, the ERK protein expression is less after 5 and 10 microgram per ml of PCB 118. If we compare the 4 hours with the 48 hours, then you can see here that ERK protein expression was less. So the results suggest that these are some possible molecular mechanisms which are responsible to trigger the cell proliferation in HELF cell protein. So in conclusion, the biphasic effects on cell proliferation was found in HELF cells treated with non-coplanar PCBs 101 and 118, which reflected as a beta-shaped dose response curve. Low concentration of PCBs 101 and 118 induce enhanced reactive oxygen generation without cytotoxicity and a cellular protective effects. In contrast, PCB 101 and 118 at higher concentration produce more RAS formation, more oxidative stress, and cellular damage as evidenced by some different enzymes. Activation of the RAS at low concentration of PCBs 101 and 118 seems to play important role in the biphasic effects of PCBs 101 and 118 on cell proliferation, and suggested that cell signaling mediated biphasic mechanisms were involved in the cell proliferation. It was found that PCB 101 and 118 showed expression of ERK1/2 protein after 4 hours, while after 48 hours the protein expression was less, indicating PCB's toxicity to MAPK proteins of HELF cells. Some future prospectus: biphasic effects of other different structures of PCBs on other in vivo and in vitro models using different endpoints such as reproduction, lifespan, enzymatic system, and proteomics should be investigated. In future, transcriptomics or quantitative structure-activity relationship analysis should be carried out to fully understand the mechanisms. Activation of aryl hydrocarbon receptor after exposure to coplanar PCBs congeners should be researched further in relation to MAPK pathway. So I have a international research networking with Professor Dr. Syed A. Hasham from Michigan State University, Professor Vladimir Strize, Mercury University, Professor Dr. Xiaofeng Xin from Chichang University, Professor Dr. Xianjin Tan from Chichang University, Professor Paramita Chakraborty from SRM University, India. Further, I am an editor-in-chief with the Springer book series entitled Emerging Contaminants and Associated Treatment Technologies. If someone from you interested to publish book, then just drop an email to me. I will be happy to accept your book proposal, and you will also receive some royalties from the Springer. Further, I am also the fellow of Society Environmental Geochemistry and Health. And this society published journal Environmental Geochemistry and Health. It is very prestigious journal and it is run by the Springer. I'm also the associate editor in this journal. If someone from you interested to become the member of this society, just drop email to me and I will be happy to nominate you as a member for this society. And this will be help you in the future. Thank you very much. And if you have some question, just feel free.
Thank you very much, Dr.
Thank you very much.
Muhammad. Yes, go ahead, please, Doctor.
Yes,
Have a question?
thank you very much, Dr. Muhammad Zafar, for talking to us about the polychlorinated biphenyl congeners induced toxicity in the human lungs fibroblast cells. I have a couple of questions. Please, what is the effect of PCB 40 and PCB 70 on HELF cell proliferation? I mean, which one is more toxic for the lungs?
Thank you very much for your excellent question. PCB 77 were more toxic as compared to PCB 40. So PCB showed 77 were more toxic to human lung cells.
Okay, so I have another question, please. On cell membrane integrity, what is most effective, PCB 40 or PCB 77?
Same, thank you very much. Same question you have just repeated that PCB 77 was more toxic to cell membrane integrity as compared to PCB.
Okay, in one slide you have mentioned that G1 first increases for then S2 G2 phase decreases after 48 to 72 hours. So what was the reason behind this? First for 24 hours G1 increased, then it showed a decreasing trend for after 48 and 72 hours.
So thank you very much for your technical question. And as you know that I was comparing two PCBs, PCB 77 and PCB 40. So you can see the results that PCB 77 first increased the G1 phase. However, with the passage of time, after 24 hours and 48 hours, these PCBs S1, S phase and the G1 phase as compared to G2. So the results suggest that PCB 77 was more toxic because they cause the injury of the cells and this injury lead to the apoptosis of the cells. When the apoptosis of the cells occur, then the expression of S and G1 phase become more. This that it suggests that PCBs 77
Okay, Dr. Zafar, thank you very much for your talk, and thanks for sharing the nice information related to your research work. And please, I would like to hand over the stage to Dr. Iman. Yes, please.
Thank you,
Dr.
Dr.
Hanan,
Hanan is
sorry.
speaking. Yes, so thank you very much, Dr.
Yes.
Zafar, for your interesting talk and also for the opportunity to invite people to publish a book with you in Springer. So if you could just please write your email below in the chat. So if the audience, if anyone is interested in co-authoring in a book chapter or in writing a book, it will be easier for them to contact you. Thank you again for the opportunity. We will move to the third speaker. We have Dr. Muhammad Sajid Khan. Yes.
Mashir, can I ask a question for Dr. Zafar?
Yes, please. Sorry, Dr. Isam, go ahead.
Okay, Doctor. Dr. Zafar, I have a small question from a microbiologist. in your introduction, you said that lung cells exposed to BCB as a vapor, correct? While during experimentation, you expose HELF cells by immersion. Is there any way to expose your cells any fumigation method instead of immersion in the BCB?
Dr. Isam, thank you very much. Actually, there was some internet problem and I cannot understand your exact question. Can you repeat the question again? There was
doctor.
some internet problem.
No
so
problem,
I cannot
doctor.
understand the question.
Okay, you understand, you listen me now? Dr. Zafar?
Yes, I can listen to you.
Okay, Dr. Zafar. Dr. Zafar,
Dr. Isam, I can listen to you. You can just drop your question to me.
Okay, Dr. Zafar. Doctor, in your introduction, you said that.
Dr. Isam?
Hey, doctor. You hear me?
Dr. Isam?
Hey, doctor. Yes, doctor. Okay. Okay, Dr. Hanan, no problem.
Dr. Isam, just type your question below in the chat box and I will read it to him if you would like.
Do you listen, Dr. Al-Mahashir?
Yes, I listen to you. I listen to both of you.
No, him. He listen you?
Sometimes his network is dropping off. So I just told him to type down his email in the chat box below.
There's some internet.
Okay, doctor, I will write a question.
Question in chat box. Can you drop a question in chat box?
Okay, doctor.
Yes, he will write it to you.
Is there any way to expose HELF cell line to BCB application? I send, doctor. I send my question.
Very much. Fumigation, maybe I didn't read any paper about fumigation through exposure of some chemicals through fumigation. Basically, when I make a standard stock solution, I use DMSO. In DMSO, the PCBs, as you know, they are hydrophobic and hydrophilic, lipophilic. So I use DMSO, dimethyl sulfate oxide. So in this DMSO, that I got the standard stock solution. The exposure to, I'm not sure. I never used this one, and I never read some paper regarding exposure of chemicals to fumigation. Can you understand? Okay. Is it fine?
Yes, it's fine, doctor. Thank you so much.
If someone asks another question, just feel free.
Okay. Thank you so much. Now we move to the next speaker. Dr. Muhammad Sajid Khan with us.
Oh yeah,
so Dr. Muhammad, Alaikum Salam. He's an assistant professor at Department of Basic Science, Imam Abdurrahman bin Faisal University. He obtained his PhD in agricultural microbiology from Al-Ghira Muslim University, India. His area of current research work includes natural plant products for their novel anti-infective activity against drug resistance pathogen, bacteria, and fungi. He has published several research papers and review papers in international journals, and he has many awards from various scientific body acknowledged. So we welcome you, Dr. Muhammad Sajid Khan, with us in the webinar, and the floor is yours.
Okay, thank you. Can I proceed?
Yes, just share the presentation.
Oh, okay, sure. Just a moment.
Okay, take your time.
Is it visible, my presentation?
Yes.
Okay. So first of all, I would like to thank the organizers of this SSDE conference for giving me an opportunity to present a part of my research work at this platform. So today's talk from my side is the topic inhibition of single and mixed biofilms of Candida albicans and Streptococcus mutans by eugenol. Eugenol is a phytoproduct that is mainly obtained from the clove oil. So basically the background of this study was focused on the oral cavity, where in the oral cavity, as we know, a number of microorganisms, including many bacteria and fungi, are there. So those microbes, they colonize the oral cavity, and they will produce many diseases like periodontitis, the dental caries, as you can see here. And also sometimes if infection becomes septicemic, they lead to the infective endocarditis, and some other nosocomial infections are caused by these microorganisms. As you see, once the infection is the establishment of infection is inside the oral cavity, it will be localized. But once they form the biofilm mode of growth, and they disseminate into the blood, it may lead to the septicemia, and also it leads to the endocarditis. So the problem from dental cavity leads to the entire body, making the disease. And in this oral cavity, actually, the microbes which are found, they are forming the biofilms. They may form biofilms either on the mucosal tissue, as you can see on the gum linings, or around the teeth, and sometimes the dental implants that we are using to treat various disorders in the oral cavity. So this picture, as you can see here, it shows a photograph with the development of biofilm around the dental implants. So the formation of biofilm either on the mucosal surfaces or on the dental implants is a great concern in oral cavity infectious diseases. And sometimes these bacteria, like bacteria and fungus, both they are from the separate kingdoms. One is prokaryotic and one is eukaryotic. These two kind of microbes, sometimes they undergo cross-kingdom interaction, and they result in the mixed biofilm formations. So once the biofilm is formed, and that is true because of the mixed condition of the prokaryotic and eukaryotic microorganisms, so it may be a highly problematic. So the problem that we want to study is that the oral candidiasis that is very common in the oral cavity, and the most of the causative agents they belong to the Candida species like Candida albicans, Candida glabrata, Candida krusei, and Candida parapsilosis. So out of these, mainly the Candida albicans is the major causative agent that is involved in causing the candidiasis. Almost 50% of the biofilm associated infections are reported because of the Candida albicans. And this organism can interact with the other bacterial oral bacterial population like Streptococcus gordoni, Streptococcus mutans, and Staphylococcus aureus. And here, this organism, Streptococcus mutans, is of major concern because this is found to be the major contributor of biofilms in the oral diseases. And this bacteria is actually prefer the low pH environment and produces the acid and other virulence factors when undergoes biofilm mode of growth. And that leads to the dental caries. And further, when the Candida albicans and S mutans, they cross-interact, they result in the plaque formation, and the biofilms are formed with the greater extension of the biofilm formation. So actually what happens here, the Candida albicans and S mutans, they promote each other in terms of nutrient requirement in the oral cavity. Means they metabolize the source of like glucose is metabolized by the S mutants by the enzyme glucosyl transferases, and then produces the alpha glucan that increases the amount of EPS in the biofilm. On the other hand, the metabolite produced after the sucrose utilization, like glucose and fructose, they are used by the candida to further promote more EPS production, and as a result, when they combine in synergistically, they produces high amount of EPS, the exopolysaccharide in the biofilm. And because of this cooperative relationship, the pathogenicity of this mixed biofilm is greatly enhanced. As you can see in this picture here, how this S mutans it can form biofilms around teeth and may lead to the tooth decay or the cavity formation here. On the other hand, Candida albicans is forming a biofilm around the tooth and the mucosal surfaces by promoting more gene expression for the hyphal development and adhesion. On the other hand, once these two organisms, they undergo cross interaction, they undergo physical interaction with each other. Chemical interaction means in terms of metabolite utilization, and they also promote the addition of each other, and overall development of EPS is increased. That results in the greater extent of biofilm formation. So the challenge that arises with the biofilm formation is that in case of the extensive biofilm, the drug resistance increases many folds. So the research has shown that Candida albicans and these S mutants, they have shown the high resistance to the antifungal drugs and to also the antibiotics. So the treatment of this drug resistant strains is quite challenging. And also in case of biofilm mode of growth, the infection becomes recalcitrant, means the recurrent of infection is achieved. So considering these all problems and challenges that are faced by the cross interaction and the formation of biofilm, and also due to the limited, because this drug resistance is actually increased because of the extensive formation of EPS that limited the exposure of biofilm cells to the antimicrobial drugs. So considering these challenges, we need to find out some new anti-infectives with the broader mode of action, and that anti-infective must target prokaryotic and eukaryotic at the same time in the pathogenic biofilms. So we have a hope with the phyto compounds, like the plants, they have been known very well since ancient time. They have the biological compounds of various chemical classes, which are known to be like anti-cancerous, antioxidant, antimicrobial, antibacterial, antifungal, antiviruses. And out of this, we focused on this compound eugenol. This phyto compound is eugenol is a major compound from the plant, that is Syzygium aromaticum. And this compound is well known. It is actually the major compound of this plant, but also found in other plants as well. So this eugenol has known antiseptic, antimicrobial, anesthetic, analgesic, antioxidant, and anti-inflammatory activities. It is also used in the dentistry as a component of the cement containing zinc oxide for sealing the cavities or sometimes filling. So eugenol that has antimicrobial activities, but the literature before they show some activity against Candida albicans and S mutans, but in a single mode of biofilm, means the biofilm of C albicans and biofilm of S mutans. But the very few studies are made on the mixed biofilm, where this compound is might be useful in eradicating the mixed biofilm formation against these two organisms. And also studies have shown, and we have also tested, that eugenol is non-toxic and considered to be the safe for human use at lower concentration. So considering this, the aim of this study was first to determine the concentration dependent antibiofilm activity of eugenol, and then to evaluate the efficacy of eugenol against the single and also the mixed biofilm formation by the drug-resistant strains of Candida albicans and the Streptococcus mutans. And further, we try to investigate the mechanism of action of this phytocompound against these pathogens. So keeping this in mind, the methods that we used was to simply isolate some clinical strains that we involved in our study. There were 11 clinical strains of Candida albicans, and we included 2 reference strains of the Candida albicans, ATCC'22 and Candida albicans MTCC'3017, and S mutans MTCC'497. And the chemicals that we use, eugenol, of course, the phytocompound as a test agent, and we use the chlorhexidine digluconate. This is the compound very commonly used in the mouthwash, and this is known to be active against both bacteria and fungi. So we use it as a positive control in our biofilm inhibition assays. And we used DMSO for making the dilution of the compounds. And the antifungal drugs and the antibiotics were also included. So the methods we used simply the determination of inhibitory concentration of phytocompounds and the drugs, and then to evaluate the efficacy of or the tendency of the biofilm formations in the clinical strains, and then the ability of the test compounds to inhibit the biofilm formation. And also we also interested to examine, means once the biofilm is formed, then what is the eradication efficiency of this compound. So the inhibition of biofilm formation, and also to eradicate the already formed biofilms. And the second, we performed some electron microscopy to examine the effect on the cell membrane and cell wall, or whatever could be the possible mechanism of action of this compound towards the both prokaryotic as well as eukaryotic organisms. So regarding the results, as you can see, when we performed the drug resistance using the MIC assays, here these strains, almost all the strains, they showed a higher level of resistance against the fluconazole, amphotericin B, and ketoconazole and itraconazole, except these 2 strains, the Candida albicans MTCC'3017 and CAJ01, they were found to be susceptible, intermediate susceptible to the fluconazole. And once we determine the MIC for the of the antibiotics against the S mutants, you can also see here there is a high level of drug resistance is found in this strain against these antibiotics. So the drug resistance is quite evident in these strains that we used in our study. And further, we investigate the biofilm formation, ability of biofilm formation in these strains. And as you can see here with the XTT reduction assay in the MTT plate, when we examined this, we found most of the strains, they were highly, they showing the strong biofilm formation and the moderate biofilm formation with the high ODs. And out of this, what we found in our study, specifically the clinical strain CAJ01 and the reference strain C. albicans ATCC'9028 and C. albicans MTCC'3017, they showed strong biofilm ability. And also the S mutants MTCC'497 showed a very strong biofilm forming ability. So we further explored our anti-biofilm assays on these strains, and we also examined the mixed biofilm formation of these strain with the S mutants. So we further focused our study to study the anti-biofilm activity of eugenol and drugs against the single and mixed biofilms formed by these 2 strains, especially the CAJ01 and S mutants MTCC'497, because these 2 strains, they showed the highest mixed biofilm formation, and our target was mainly to explore the compound against the mixed biofilm against the mixed biofilm formation ability of these 2 cross-kingdom interaction. And if you see, once we determine the MIC in case of planktonic stage, and also in the biofilm stage, that is a sessile MIC. So what we found, if you see for the antibiotic against the S mutants, the drug resistance, the MIC increased almost fourfold here. And if you compare for antifungal drugs for these Candida strains, the sessile MIC is also increased almost 64-fold here, and greater than, it was like highly increase in the SMIC was observed. But once we compare it with the eugenol, what we see, the eugenol, in case of eugenol, the MIC was, the sessile MIC was just twofold increase we observed in the planktonic and the biofilm stage. And for the S mutans, as you see, there was no change in the MIC in the planktonic and in the sessile condition. So further we examine or evaluated the efficiency of this eugenol and eugenol in inhibiting the biofilm formation. So we took the sub-MIC concentration for eugenol and like 25 microgram per ml, 50 microgram per ml, and 100 microgram per ml, and we use chlorhexidine digluconate as a positive control here. So you can see at the sub-MIC levels with the increasing concentrations, the efficiency of biofilm formation is decreased significantly by the eugenol, and against the single biofilm of candidialbicans and also against the single biofilm of S mutans. And the same pattern is also observed against when the mixed biofilm was allowed to form. The eugenol was quite able to also inhibit the mixed biofilm formation at sub-inhibitory concentration. And further, once we try to evaluate the eradication of biofilm formation, we perform the viability of the biofilm cells, and after the biofilms is formed and then exposed to the sub-inhibitory concentration of eugenol. Here we considered the untreated as a negative control, and the digluconate is used as positive control, and these are the sub-inhibitory concentration of eugenol that were used. So here we also see, if you can see, compared to the untreated control, in case of candidialbicans and S mutans, what we observed, the viability was decreased from log CFU, that is 6.3 to 4.864 in case of candidialbicans at 200 microgram per ml of the eugenol. And at the same concentration, in case of S mutans, the log CFU count was reduced from 6.4 to 3.875. This says the significant reduction in the biofilm mass once it is exposed to the eugenol. And the same when we allowed the mixed biofilm to form at the same concentration, 200 microgram per ml, what we observed, the log CFU was reduced from 6.3 to 4.866 for Candida under mixed condition, and 5.3 for S mutans under mixed condition. So this is also a significant decrease compared to the untreated control. So these studies reflect that eugenol is quite capable of inhibiting and eradicating the biofilms either single mode or in the mixed mode by the candidialbicans and S mutans. And further, we performed scanning electron microscopy. As you can see, at the sub-inhibitory concentration, the treatment was given to the candidialbicans alone to form biofilm, and the S mutants to form biofilm alone, and then under the mixed conditions. So as you can see in the untreated panel, you will see they show the developed mature biofilm here in single condition of candidialbicans, single condition of S mutants, and the mixed condition where is the thickest amount of EPS is quite observed in the mixed biofilm. And once the eugenol was given at 50, 100 microgram per ml, and this is the digluconate was given at 200 microgram per ml, and you can see this is with the increasing concentration, there was a distortion in the biofilm was observed. The EPS is also decreased. The amount of EPS is decreased, and also we see the alteration in the cell membrane and cell wall structure. As you can see here in this picture, this is the treatment of candidialbicans at 100 microgram per ml of eugenol. It shows distortion of the E cells, and here you can see the permeabilization of the E cells. On the other hand, the S mutants, they also show the distortion in their cell membrane and the cell wall. And under the mixed condition, if you see both the Candida and S mutants, they show shrinkage in their cell envelope, in cell membrane. So overall, when we perform this scanning electron microscopy, what we observe the shrinkage and distortion of the cell in the S mutans biofilm. And also we observe disturbed cell wall structure in the mixed biofilms. We also got lysis and release of intracellular material. So this preliminarily indicates that there is the target of the eugenol towards the cell membrane synthesis or the cell wall synthesis here in the Candida albicans and in case of S mutans. So overall, our study says with these observations of electron microscopy and the ability to eradicate and inhibit the biofilm formation, the eugenol is found to be a great anti-biofilm compound, which is going to eradicate as well as inhibit both single as well as mixed biofilm, and it shows multiple mode of action where it is targeting the cell wall and cell membrane. So it could be a great choice to use in eradicating this mixed biofilms in the oral cavity, especially for the Candida and Streptococcus mutans. So further, we are aiming to investigate eugenol. What is the effect of eugenol on the expression of the genes in Candida biofilm, like addition genes ALS3, EFG1 for hypha induction, and the virulence factor that is SAP5 and HWP1, means whether the eugenol is going to suppress the expression of these genes. And Streptococcus, in case of Streptococcus, we are further trying to test that whether they are going to suppress these two gene in when it is exposed to the eugenol. And further, once we confirm with this molecular investigation, we can also go for like in vivo trial to come up with the possible integration. Means we can say the eugenol could be further used to be in the modern phytomedicine, and sometimes this eugenol can be used in combination with the already existing chemotherapeutic agent, because this eugenol is most of the studies have shown it is showing the synergistic interaction with the antibiotics and antifungals. So it could be a good choice to either use as a single therapy or in the combination therapy as well to eradicate the biofilm associated infections, especially in the oral microflora. And these are the references that we have used for our studies. And thanks to all for listening my work here.
Thank you so much, Doctor.
Yes, thank you.
Thank you so much for your presentation. And if anyone has a question, just please type them.
Yes, Dr. Mashir.
Yes, go ahead, Dr. Ethan.
Dr. Muhammad, you have got some pictures for Candida. So my question is how you prepare your Candida for scanning electron microscope. Did you use dehydration, alcohol dehydration? And what is the type of coating for your sample? Is it chromium or gold or other?
We used dehydration for this and not dehydration exactly. We are using this scanning electron microscopy for once we prepared, like on the cover slip, we prepared the biofilm. And after that, of course, it has gone for dehydration synthesis in the preparation of the electron microscopy. And also the coating, it was, I think it was gold or something.
Gold?
I exactly don't remember because it was done with some other stuff with the help of Ben. We did it. So exactly, I don't remember it.
Did you make lyophilization for this sample?
No, we did not make lyophilization. We take out the suspension and then we take it to the scanning electron microscopy.
Okay, doctor. Okay. I noticed that you all the time you study the MIC. Why didn't you study the MBC? Why not MBC?
We also studied MBC, but only because our aim was to examine this efficiency at the lower concentration, means sub-inhibitory concentration, not at the MBC. MBC, it is going to kill the organisms. So what we, and at the lower concentration, we are trying to get, means it is also non-toxic, one thing. And second thing is that at the inhibitory concentration, if it is something giving you result for the antibiofilm, that is better, because MBC is going to create more resistance level. If something is at any inhibitory concentration, it's good as antimicrobial efficiency.
Yes. Yes, doctor. Yes. Okay.
Yeah, thank you.
Thank you again. Thank you for your presentation. And we will move to our last speaker, Dr. Jacob. Are you here? So we will wait 5 minutes for Dr. Jacob, and meanwhile we will have the video. Isaac, can you start the video? Yes, hello, Dr. Jacob.
Hi, can you hear me?
Yes, I can hear you loud and clear. So,
Hello. Yeah.
first of all, thank you for coming. And I will
Thank you
just
very much for
introduce
inviting
you
me.
to the audience. Dr. Jacob Gayali is an assistant professor of physics and the leader of quantum group at the University of South Florida. He focused on electrical transport in DNA molecules and computational studies of semiconductors. He received his PhD in physics from Texas A&M University in 2016. And he has many published papers in high impact factor journals and others. We welcome you with us, Dr. Jacob, and the floor is yours. If you can just share your presentation.
Okay, is that working now?
Yes.
Oh, good. Should I begin?
Yes, go ahead.
Oh, so first, thank you for inviting me for this very nice seminar conference or webinar, I guess one would say. Yeah, today I would like to talk about some of our recent research on multi-band topological semi-metals for next generation devices. And yeah, of course, as she said, my name is Jacob Gels, and I'm an assistant professor at University of South Florida. And some of this work is actually published and actually no longer archived. So, but you can still find it on the archive, and there are also some other works in here that I will discuss. So what is South Florida like? This is actually the Interdisciplinary Science Building, which is a mixture of physics and cell and molecular biology, which is located at the University of South Florida, which is actually closer to Central Florida here in Tampa, but it's a composition of 3 campuses that make up the university. So one in Tampa here, one in St. Petersburg, and one in Sarasota, which is, home to some of the nicest beaches in the world. Yeah, which I'm very happy to be a faculty of. Some of the collaborators especially for this work, not all of them, but some of the main collaborators are Professor Dr. Felsler, who does the experiment and led by her member Stanislav Marko, and Jan Slun, who's no longer at Max Planck Institute, and some of the students that also helped in some of the projects. And then at the Max Planck Structure for Microphysics was Mazhar Ali, but who is also recently moved, and Elena Deruvino, who was a postdoc there, who moved, and then also Michael Gutois, who is also a postdoc at University of Liverpool. So the quantum chirotronics group at USF is composed of several members. We have a postdoc, Dr. Bushra Kabir, and she's actually has helped a lot with a lot of the calculations, and we're finishing up a paper on some of the work that we will discuss today. And then we have some graduate students and also an undergraduate student who's actually helped a lot with also some of the simulations here in the magnetic regime, Cole Gibson, and some other students also are shown here. So what does our group focuses on? And at the moment we're growing, and so we focus on a few different regimes of magnetic and topological and chiral materials. And so this includes what's called a nanoparticle. And what we see here in these kind of cases is as one shrinks the size, you can kind of determine the electronic structure at different scales. We can also control the exchange parameters based on the size and also the type of materials, which leads to what's called the skyrmions. And sorry that the quality is not so good here. I don't know if it's the same on your end. And then we also have the materials that we focus on today, these electronic structures for magnetic materials, which have these very interesting set of topological bands. These usually come from what's called a wow semi-metal. And what's interesting about them is they have some sort of topological surface that can be manipulated and used for electronic devices. So this can be used to increase battery power, and also use things such as solar cells, and also maybe be able to use in next generation topological quantum computing, but this is much further in the future. And so to continue, some of the actual materials we'll be talking about today is mainly 2 materials, this chromium platinum 3 and this manganese bismuth. And there's some other materials such as platinum manganese antimony, or platinum manganese gallium, and some others that are very similar that we'll briefly touch on. And what's interesting about them is they have what's called this electronic structure, which is the way the electrons behave and momentum space, or how they interact in the crystal environment, which is very interesting compared to normal metals. And I will go and explain this. The main part here is that usually these magnetic well semi-metals are reduced to very simplistic systems with only 2 electronic bands, which I'm showing here, many electronic bands. And here we want to look at something very complicated, these very complicated metals, because they hold a lot of interest that can be quite beneficial for future technologies. And so with this very complex system, today we'll kind of go through and try to disentangle it. So to start, one thing that we can talk about for these for metals is what's called, for magnetic metals is what's called the anomalous Hall effect. And this is a general graph for the anomalous Hall effect, and I will explain in more detail exactly what the anomalous Hall effect is. So what we look at here is the conductivity of the material by either changing temperature or the impurities in it. And you get into 3 different regimes when you compare it to the anomalous Hall effect on a log scale, yeah? So there's one regime which is called this localized hopping, where you usually have a very low anomalous Hall effect, but it scales something like with the conductivity to 1.6. And this is because on a log scale that this scaling is linear here. And then on the other regime, you have what's called the skew scattering regime. And this usually scales to something like sigma to the 2 or sigma to the first power. This is actually a material we studied very heavily, was this Canadian vanadium antimony. It has been a very nice paper where the anomalous Hall effect scales drastically. You can actually reach even some newer regimes. And what we're going to focus on today is the anomalous Hall effect in these metals and this intrinsic regime. And within this intrinsic regime, which I'm going to zoom into shortly, you have different types of metals, such as your iron type metals, your cobalts, or your nickels, which are very common metals and quite well understood. And when you go really deep into this, you'll start seeing that these topological metals exist right on the border here. We're going to try and disentangle what happens there. So let's zoom in onto that. This is some results that, so this is kind of the shocking result that we got for a particular material, this chromium platinum 3. These are all experimental thin films that we wanted to compare with, where the previous was a mixture of bulks and thin films. And so we have that particular case of iron, which, shows this intrinsic regime, and it's really called intrinsic because for the most part, the anomalous Hall effect on this log scale is constant as a function of the diagonal conductivity, or the longitudinal conductivity, which is shown here for these simple metals such as cobalt. Here you have nickel, here you have iron, and gadolinium you have here. And so one thing that you see is that these topological metals, which we will also go into more in detail, on the left side here, they usually end up on the left side here because they don't have many electronic bands and they're not metals like we're used to. So they're what's called semi-metals. And what we've seen for the most famous of these topological metals, which is cobalt-3 tin sulfur and cobalt-2 manganese gallium, the thin films don't do as well as the bulk. And the theory, which is this red line for the cobalt-3 tin sulfur, does not is not so well in agreement with the thin films. And with this chromium platinum 3 with different stoichiometries and different variations from the experimental groups, we see that the theory, which I calculated and work on, does a very good job at predicting and capturing the effect of these materials. And there's a particular reason why, and this is what we're going to go through in the talk. And if there's any questions, please stop me and ask me if it's possible. I know sometimes it can be a bit different with webinars. Oops. Oh, sorry, today. Give me a second. This is still, you can see my screen, hopefully. To give an outline, I thought that I wasn't sure exactly at the level of everyone, but to make it as open as possible, I will give discuss this idea of band structure from the beginning. And then I will talk about this anomalous Hall effect in different regimes. And so I really go into detail there on what it means for the anomalous Hall effect. And then I'm going to go into probably the strongest part of the talk is the simple model with multiple bands to really, that's probably the take-home message I want everyone to get here. And then I'm going to go into some of the actual systems that we have, this cubic, which I call the cubic XPT3 compounds, and the work that we're working on now and that we published already. And then I will go into the magnetic textures and this WoW manganese bismuth. And this is quite new results that were just published in the last few months or so. And then I give a summary and you can also ask any questions at that time. Sorry, yeah. So the band structure. So we start with a quantum state, which is, this wave function for a free electron, which is described by this parabolic shape where you have some h bar square, which is Planck's constant, the momentum state squared divided by the mass of, in this case, an electron, and you can have some average effective potential here. And what happens here when you add this potential, if it's periodic, you can open up an energy gap due to the electron wave functions canceling on the boundaries here. This energy gap is the way everyone has thought of materials up until recently in the last few decades, and this Nobel Prize that has come out in terms of topology in 2016. And so the idea here is that with the energy gap, you can determine materials if they are metallic, semiconducting, or insulating. Yeah. And what that determines is if you start filling in electrons into this, what this is here now is a simple band structure where we determine the energy based on the momentum of the electron. If you fill it halfway up in this red band, then you have a metallic state because it's easy to excite the electron higher up in level. But once you get into this forbidden energy gap, you come to an insulating or semiconducting state, depending on the size of that gap. And this has had, tremendous impact in different technological systems such as computers and sensing, for the most part. And the size of this energy gap is determined by the potential, which is usually about twice the size. So then we can go to something, a little bit more complicated, and we can look at a free electron, and we put it a potential that looks, depending on the type of the crystal shape that you have, that the atoms arrange in, you can have different symmetries, and these symmetries have a global effect on the electronic structure. So with a BCC, you have a particular band structure that follows with it as compared to with an FCC, which can be quite different. And this will give very different dynamics of the electrons. And so here, one thing that changed recently is that people started to bring in what was known as relativistic effects. Once you start filling in those different lattices with potentials, you can put a very heavy potential. And what you would need now, instead of the Schrodinger picture we had, you need to bring in this Dirac picture, right? Where the speed of the electrons can be on the order of the speed of light, and the mass can also includes this rest mass. And this is this Einstein, the famous Einstein equation. When there's no momentum, then it's just E equals mc squared. And that mc squared there is that gap that I was talking about. And here now, instead of those two states you have 4 states here that also depends on the spin of that. And this model actually, when you have no mass, explains graphene, which is this wonder material that came out recently. Which is a 2D material of carbon atoms that lies flat like a chicken wire, and it has this linear crossing of the band, this electronic structure, right? And what's interesting about that is that now you're talking about electrons, but they're the way we model them and the way we look at them theoretically is it behaves very similar to light, right? And so these electrons move extremely fast, they move around defects, and they behave like light particles. And this material is also quite interesting just because it, it's transparent at one layer, and it has it's very durable, and so people really liked it. And the reason for this is not only in physics, it also has explanations in chemistry, where the bonds that make up this system is due to these carbon atoms that have these different types of orbitals that hybridize and produce this sp2 bonds, and this becomes like a semiconducting insulating state. And then you have this pz bond that's left over, and this is like this metallic state where these electrons are able to move with these near light speeds, 10% or so, yeah. And this was predicted by, first shown by Andrew Geim and Constantine, and they actually got the Nobel Prize for this. And what they did was, just taking a piece of pencil, which has this graphite, and they would take peel off pieces of the single layers of the graphene with Scotch tape, which is, I think, one of the coolest experiments in the last few decades. And so what really determines these things is really the symmetries of the system, right? So this 2D graphene, which also could be rolled up into nanotubes and things like this, and changes the symmetry of those electronic structures. It's all determined by those symmetries. And one of the symmetries that we can break some symmetries in that Dirac Hamiltonian. One is we can break inversion symmetry. So, you know, if it has, you can take out this left to right inversion symmetry. And what happens here is this is just showing a mirror inversion. And what happens is you break this Dirac cone that I shown earlier into two cases of what is called two Wow cones, right? And so what's the difference between these is that they have opposite spin chiralities. And that's partly the name of my group, with this quantum chirotronics group. These chiralities actually have some meaning, and they have some connection that can't be easily broken. And then also the other case is the time reversal symmetry, which is this magnetic effect. If you break that, you can also separate this Dirac cone, which is easily seen because of the spin, into two different Wow cones here. So this really determines all the Wow semi-metals, which you need to somehow break these two, either or both of them in some way, or just one of them, to actually get a picture of this Wow type Hamiltonian. So next part. So now that you guys are all experts on the band structure, we're going to focus on the anomalous Hall effect and these different regimes that we kind of discussed earlier in the beginning, and go a little bit more into detail here. So what is the anomalous Hall effect? This starts off also very simple and this started out very simple, much longer ago than the band structure theory, and it started out in the late 1800s, where Edwin Hall took a piece of metal and he applied a magnetic field, and then he also applied an electric field, and he calculated something very simple like the Lorentz force, and he saw that, you know, that the electrons and holes would bend to one direction and create a voltage perpendicular to both the magnetic field and the applied electric field. And you can measure that, and, you know, 1 year later, he took a magnetic material and found that this effect was gigantic. And so there he wanted to, and for the last almost 100 years, people have been trying to figure out what that is. And you really need a good quantum picture to actually understand the second effect. The first one, mostly determined by classical effects. You can add some quantum effects in there, but for the most part, it's well understood from the classical regime. And so this is the anomalous Hall effect, the second case, which is started in magnetic materials, which doesn't have to be metallic or not, but it's magnetic. And so to really start about that, I would rather talk about what's called the non-magnetic regime. And so pretty much what this is, the non-magnetic regime, you have some electronic density states where you have electrons that spin up and electrons that spin down, and they're equal in amount. And as you apply electric field, what will happen is, due to that the Dirac effect, you have what's called spin over coupling. Every material has it, just in different degrees of size. It splits the electrons across, and you have what's called you have a no net current transverse, but you have a spin current that accumulates on both ends. And this is a very interesting part, because it's very difficult to measure this experimentally. And you can calculate this from the electronic states and the currents and the velocities that come out. And so then once you start to apply a magnetic field, you change the population of the spin up states compared to the spin down states. And now you have more electrons or less electrons on the side, and you can actually measure a voltage here. So that's what Edwin Hall measured in 1889. And the spin Hall effect actually was not really measured to 2004, I believe. 2004, 2005. And they measured, the calculations are very similar in the same way. One is just a spin current, one is just a net voltage that we measure. And so let's talk about what happens with this anomalous Hall effect of these different regimes. It's best to talk about this in terms of semi-classical picture, because it gives the most accurate description in terms of an easy and intuitive to understand. Yeah. And so the first regime where I showed things were, you know, moving quite large and linearly. This is called the skew scattering regime. This is where either you have a magnetic system and you put an impurity in your system, or you have a system and you add a magnetic impurity. And what will happen is your spin electrons will very simply scatter off that impurity, and because of the spin, will go different ways. And then you can see this in things like Magnus effect, where if you spin the ball, it will, the way Cristiano Ronaldo kicks a soccer ball could either go to the left or right, and this is a very similar idea there. And so that's what we saw in this potassium vanadium antimony, where we also added some magnetic texture there, and it was able to really supercharge the electrons there to get to very large values, some of the largest seen in experiment or theory ever. And the idea was that, okay, yeah, you can have this clean regime where you have this magnetic system and impurity, or you can have this other case, this dilute Kondo system where you have a magnetic impurity. However, both of these rely on impurities, and in this case, what we relied on here was this changing of the magnetic texture as the impurity, but not an actual atomic impurity. And that's why we were able to get to very clean and very large anomalous Hall effects, which is this potassium vanadium antimony. And the results you can find in the paper more if you're more interested, or ask me. The other regime is called the intrinsic regime, and this is the regime I've been talking about the most so far. And this is where you have some what's called Berry curvature, and this is due to the electronic band structure, which nominally is the largest effect in these systems, especially in the intrinsic case. And then lastly, you have what's called the side jump regime, and this is purely a quantum effect where you add some phase to your system when there's an impurity. And this is really independent of the impurity densities, but more important on the strength of the impurity. And you can classify these, experimentally or theoretically by separating them based on the conductivity. So let's go to the simple model with bands, which is, probably the part that I will spend the least amount of time, but the part I would say the most important. And so we don't have to spend too much time here on the Hamiltonian. We can look at a Hamiltonian simple model where we have some Dirac type physics here, this relativistic spin orbit coupling, which couples the spin. These are the Pauli spin matrices to the motion or the momentum of the electron in different directions. And then we have the magnetization, and we have the normal kinetic energy hopping of the electrons that is, coupled to this the spin Z component. And what happens is, and this simple model just relates directly to this case here, where, you have this chirality that pops up, and you get this anomalous Hall effect as well, pretty well defined and very easy. And I can play around with this. I can change the sign of my spin orbit or the sign of my magnetization, and I can flip those bands, change the chirality or invert it, and I can change the sign of the anomalous Hall effect. Now, one thing that hasn't been really seen until down to this paper that we had was that you can actually, combine these things. Many times people say, yeah, look, I have a lot of these crossing points in my material. Why don't I have a very large effect? And so we went into detail with this into these multi-band systems. And so you can either cancel or enhance these materials based on how you add in the chirality. And so when you add them together, it can cancel out, and when you also add them together where it enlarges. So, and there's many different cases. The prime examples for this is this cobalt 310 sulfur, which is one of the first wild semi-metals found, and you had this very large anomalous Hall effect, but you had a very simple band structure, and you had 2 wild points in the system. Yeah, so everything was quite straightforward and easy to understand. And then you also at the same time had this cobalt 2 manganese gallium, which is also a simple band structure where you only have 2 bands, and it was very nice because it was very easy to understand, and also showed, such effects as thermal effects and things can be found. And there were a lot of experiments on this that, showed that, as you decrease these materials, you can create a quantum anomalous Hall effect, which is also done from our group in collaboration with Claudia Fessler. And so in the next case, we had another material very similar to cobalt-2 manganese gallium, was cobalt-2 titanium tin, and we knew there were a lot of crossing points, but the anomalous Hall effect was pretty much 0. And what happened here was we saw that it was really due to this cancellation of these bands where you have these different chirality effects shown here, where you can see it in this Berry curvature picture. So let's move on now to the cubic XPT3 compounds, which is that last part I didn't show yet. And here, this is this cubic compound, which is very interesting because it's quite well known and was used for magnetic optic recording, where at this corner you have some transition metals such as chromium or manganese or vanadium, which I actually show here. This is theoretical calculations, and then you have this platinum here. And because of this magnetism, although the structure is cubic, the symmetry of the electron sheet is actually closer to tetragonal, and you have some magnetic moment. I show as a function of the composition of manganese and vanadium and vanadium. Here I showed some quickly. I showed some experimental just of the XRD of the thin films with different compositions, showing that it has a very high crystallinity, and we can also see that that it's magnetic and it has a little bit of hysteresis in there. And then we even can look at the resistivity or the inverse of the conductivity here for the different compositions, and also the magnetoresistance, showing that these materials were very well prepared. It's a very good job by my colleague Anastasios Marco in Dresden. And also we look at the end plane and outer plane magnetoresistance. Before we finish up the main thing here, I just want to compare all 3 band structures of the cobalt-3 tin, which is the one of the first Weil semi-metals, which is just these 2 bands here. It was very nice and easy to understand. And then you have the cobalt-2 manganese gallium. And now I'm going to show you the chromium platinum 3, which was just a mess for us when we started, right? How do we understand what's going on here? There's just so many bands. It's not pretty to look at. And so we started to disentangle each band by itself. And what we found was that there were 2 sets of bands. This is in a vanadium platinum 3 case. This two blue, this 2 red, that contribute to all the topology for the most part across the regime. And this is the anomalous Hall effect you can see, which is mostly dominated by the red and blue. And then as you change it from vanadium to chromium, you can see the bands change a little bit, but it's still the same 2 bands that give rise to topology. And as you go to the manganese part, it's still the same 2 bands here. It just changes only a small amount. And here I just show the anomalous Hall effect, as a total, and then the dashed line shows the contribution from the rest of the other bands. And this is how I calculated it. So let's continue. So as we change from vanadium to chromium to platinum, we can change the magnetization. What we see is that the magnetization does not relate directly to the anomalous Hall effect, but we can really change the anomalous Hall effect, shown in blue here, by orders of magnitude. And also at the same time, we can even change the anomalous Nernst effect, which is this effect that is exactly the same as anomalous Hall effect, but you get a temperature gradient out instead of a electric field. And this here shows you what, before we add this direct spin-over coupling, you have what's called these nodal lines, where these are, instead of a point that touches, it's a whole line and momentum space that are conjoined and that are degenerate. And the next slide is, is the Berry curvature and showing where those degeneracies open up with spin-over coupling, or this Dirac relativistic parts of the Hamiltonian. And then I want to just really quickly compare it to the experiments, the results. So this is the anomalous Hall effect calculated from the experiment. This is actually the anomalous Hall resistivity, and one inverts this to get the anomalous Hall conductivity, which is shown here for different temperatures. And this is how one changes the conductivity as a function. And what we saw is we can separate those different, skew, scattering, and side jump, which I showed earlier, to these different components. And this is all the experimental work here, and it was actually came out very nicely. And that's how this component here, this intrinsic parts, what we plotted here, yeah? And then we compared, this is the graph I showed you earlier, I think we don't need to spend more time on. Yeah, I think I have about 30 seconds, correct? Or how much time do I have?
About 30 seconds.
Okay, yeah.
Okay.
Yeah, so this is the graph I went over a lot. I think everyone followed. I just wanted to very quickly talk about manganese bismuth, which is another interesting material, different shape of a compound. It has a hexagonal crystal structure and is made of manganese and bismuth. Both are very interesting because of the spin-over coupling and the magnetism. And they lead to another chiral effect, which is called a skyrmion. And these are different types of skyrmions. I won't go into much detail here. And then I will skip this slide because I think I have not so much time left. And I'll skip this one also, just to get to the main results of the material. Sorry for that. So the main results here is that what this compound does, it combines two chiral effects. And this is the magnetization phase diagram as a function of temperature, where and the bulk samples, you go from a saturated ferromagnetic and polarized state and to a conical state, and you can even stabilize the skyrmion bubble, which I've shown before. But which is even also more interesting is that the band, it also has these topological bands that exist. And this is my theory directly on top of the experimental work. And one of the things that comes up here is that it has some of the largest values for mobilities for magnetic materials seen, and is actually much larger than this cobalt 310 and this cobalt 2 manganese gallium that I shared up with you earlier, which were some of these first interesting materials. And these are most of the other materials. These are topological materials. You can see the effect here in this case is more than double or 4 times anything else, and these are most materials here. So that's pretty much it. The summary is here, of everything we went over. I hope it was very clear, and thank you for your attention. And if you have any questions or would like to visit Florida or anything, please email me. I'll be very happy to continue discussion. I don't know if you guys are recording, but here's a summary of all the references. Some of them are published now, and some of them may not just be yet. But if you have any questions about it, you can definitely email me if you need that slide. Thank you.
Thank you very much, Dr. Jacob. Anyone has a question?
Yes, please, Dr. Hanan. Can I
Yes, go ahead, please.
ask Dr. Hi, Dr. Jacob. Good morning. Indeed, I have a very important question to you. How is the weather in Florida today?
Oh, it's very yeah. I can show. I don't know. Maybe can you see my screen? Should I stop sharing? I can show you very quickly. Maybe you can see there. It's very beautiful.
Wow. Stunning weather.
It started quite early, but by
yes,
now
it's
the sun is
7
coming out.
7 a.m.
Yeah, 7:30 or so.
Yeah.
Yeah.
Okay,
Yeah.
I'm joking, by the way. So, Dr. Jacob, you are using the bismuth, and experimentally, we know that the bismuth, this semi-metal, when we use it, for example, with other materials, for example, the gallium arsenide, the indium arsenide, semiconductors, for example, by MBE or MOCVD, the bismuth has a very important physical property, which is the surfactant effect. Indeed, I want to know, in your calculation, do you introduce this physical property in your calculation? Can you introduce this surfactant effect, which experimentally it greatly alter or affect the main or the overall property of the final material or the final alloying?
I'm sorry, I'm not the smartest, but can I ask what the surfactant effect is?
The bismuth. Yeah, it has a
Yeah.
surfactant effect, okay? Because I used to work with bismuth. By MOCVD, we make a semiconductor, and we introduce the bismuth to ameliorate the properties of those materials. And we face a problem, which is the surfactant effect of this material. It bubble up to onto the surface of the of our alloy. Do you introduce this parameter in your calculation?
So in a short answer, no. One, because for the manganese bismuth here is actually something. I'm not an experimentalist, of course, but I know it was very, very tough to grow. I remember I was particularly looking for something. I had gone through many materials, and I was looking for something special because I wanted something that would combine something like manganese, which has 5 electrons in it, and so that usually wants to be antiferromagnetic.
We cannot reach even 0.7%. We cannot exceed this value experimentally.
Well, I think it's just particularly this compound. So yeah, so bismuth is always, very. I've heard it's always very complicated to grow with bismuth. But I think this one, that's the one time I know just for this manganese bismuth, it came out very nicely. So it was not. I think it was more of luck than anything, pure, ingenuity there. But it was just we were searching for a particular compound that combined almost antiferromagnetic with lots of spin-over coupling that comes from bismuth.
Do you introduce usually in your calculation such kind of that problems in your calculation?
No, because I'm much more interested in the electronic structure. So if it has a stable energy, I'm okay with it. But I mean, in this case, I would
So
have
you
to
work
go
at the
to
safe side.
Yeah, I work on the safe side. I mean, I would look at the cost of energy of the system, and if it's okay, then I'm okay with moving on. But I'm usually not doing molecular dynamics, and that's what you would need to actually, if I understand correctly, to actually capture the surfactant effect. And those calculations, those guys are much more ingenuitive than I am, and they spend a lot more time to do those calculations, a lot more computers. So I like to look for a little bit more fundamental, something that I can finish in a day in terms of a calculation.
Okay, thank you very much, Dr. Jacob.
Thank you very much for the very nice question.
Thank you, Dr. Iman, for this question, and thank you, Dr. Jacob, again for the interesting presentation. And to all of the presenters today, we thank you very much. This is the end of the session 2. It was my great pleasure to open the first day of the webinar and to chair the 2 sessions of the Bioscience Ecosystem and Environment. And we hope to meet you all again tomorrow for the material science and the chemistry sessions. Thank you very much.
Thank you very much, Dr. Hanan Al-Mahashir.
Thank you.
Thank you for this nice session today. We enjoy with you, really.
Thank you.
Thank you.
Bye.
Bye.